Bengkulu berkomitmen menjadi pusat pengembangan ilmu proteksi tanaman tropis yang bereputasi internasional, menghasilkan lulusan yang kapabel, adaptif, dan siap berkontribusi dalam pengelolaan pertanian berkelanjutan berbasis sumber daya hayati.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Program Studi Proteksi Tanaman atau PSPT
nanti ibu yang share screen sendiri.
Ya nanti saya yang ya. Oke.
Ya, oke.
Senang sedikit sendiri.
Iya, iya.
Mimi,
Ya,
maybe we can start now. It's already read for 20 minutes.
ya, ya. Oke, Pak, bentar.
Sama backgroundnya gimana anunya Bapak biasa tuh model barunya, nah gimana nggak muncul ya? Hah? Pakai apa ini?
Cek satu,
Oke.
cek tes,
Lalu untuk
tes,
background
tes.
di sini gimana ini? Oh nggak ada ya?
Ini Pak. Oh, ada nih international lecturer kita nih. Pak Dr. Henry.
Terima kasih Pak.
Biasa. Di mana sekarang nih Pak? Di kampus.
Di kampus, di
Oh,
Unip
di
ini.
kampus.
Ya. Ini lagi setting ini.
Saya lagi setting background saya supaya nggak terbalik nih gimana.
Oh, di mirror.
Saya pakai tablet, saya nggak ketemu.
Oh, iya. Kalau tablet mah lain. Luar biasa itu.
Ini kayaknya.
Kita yang nggak punya tablet, Pak.
Jangan cek itu, Pak WR.
Bapak udah ini. Pak video ya dan Pak.
Sudah betul itu background Pak Dekan.
Ada ini.
Pak Henry yang belum. Backgroundnya masih apa tuh?
Nggak, kalau saya lihat saya sendiri terbalik gitu.
Tapi yang betul Pak, yang di sini betul. Yang tampil.
Oke, berarti di saya ya, kamera saya ya. Oke.
Ini.
Baju lagi ya, Pak?
Ya, oke. Ini Bapak lagi. Oh, udah. Sudah. Terima kasih ya. Ini kecil backgroundnya, supaya besar gimana? Mm-mm.
Baik, kita mulai Bapak Ibu.
Ladies and gentlemen, to open today's event, we proudly present Tari Persembahan, a traditional welcoming dance from Bengkulu.
This dance symbolizes respect and gratitude to our honored guest, Tepak Siri. A key element signifies a warm welcome and the hope for harmonious relationship. Please enjoy Tari Persembahan.
Distinguished guests, ladies and gentlemen, we will now proceed with the singing of the Indonesian national anthem, Indonesia Raya. We kindly invite all participants to join respectfully from their respective location. Assalamualaikum warahmatullahi wabarakatuh. Good morning, distinguished guests, keynote speakers, participants, ladies and gentlemen. May peace and prosperity be upon us all.
We are honored to welcome you to the second International Seminar on Plant Protection 2026 with the theme Horticultural, Plant Pests and the Diseases. This seminar is organized by the Department of Plant Protection, Faculty of Agriculture, University of Bengkulu, in collaboration with the Indonesian Entomological Society and the Indonesian Phytopathological Society.
We would like to acknowledge the Rector of University Bengkulu, represented by the Vice Rector for Planning and Cooperation, Professor Asar Muda Lubis, SSI MSc PhD, the Dean of the Faculty of Agriculture, Professor Dr. Insinyur Suharyanto SPT MSI, the Head of the Department of Plant Protection, Dr. Mimi Sutrawati SP MSI, the Secretary of the Indonesian Entomological Society, Dr. Misyahrawati SP MSI, the Secretary General of the Indonesian Phytopathological Society, Professor Ani Widi Astuti SP MP PhD, and the Chair of the International Seminar on Plant Protection 2026, Organizing Committee, Dr. Yeni Sari Asi SP MSc.
We are pleased to welcome our keynote speaker, Dr. Cheng Fang Hong from National Chung Sing University, Taiwan, Dr. Sofana Ramon from the National University of Battambang, Cambodia, Dr. Norida Mazlan from University Putra, Malaysia, Professor Agustin Zarkani from the University of Bengkulu, Indonesia, and Professor Amelia Nicolas from Central Bicol State University of Agriculture, Philippines. We are also honored to welcome our distinguished invited guests and participants.
Let us begin by expressing gratitude to Allah Subhanahu wa Ta'ala for His blessings and mercy, which enable us to gather online for this international scientific forum. To start the event, let us listen to the recitation of the Holy Quran, to delivered by Zaki Nur Atif, who respectfully invite the reciter to begin.
Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Innallaha falikul habbi wan nawa' yukhrijul hayya minal mayyiti wa mukhrijul mayyiti minal hayyi. Dzalikumullahu fa anna tu'fakun. Falikul isbah وجعل الليل سكنا والشمس والقمر حسبانا
Bukan ini kampusku ada seminar internasional jadi nengok. Iya pembacaan ini Jer. Iya.
Beriman sekali.
Lah biar berkah cuy.
Berdoa aja.
وهو الذي أنشأكم من نفس واحدة فمستقر ومستودع قد فصلنا الآيات لقوم يفقهون وهو الذي أنزل من السماء ماء فأخرجنا به نبات كل شيء فأخرجنا منه حدير نخرج منه حبا متراكبا ومن النخل من طلعها كنوان دانية وجنات من أعناب والزيتون والرمان مستبها وخيرا متسبح انظروا إلى ثمره إذا أثمر وينعه إن في ذلكم لآية لقوم يؤمنون صدق الله العظيم. Assalamualaikum warahmatullahi wabarakatuh.
Salam.
Thank you for the beautiful recitation. May these secret phrases bring blessing to our seminar. Ladies and gentlemen, we will now proceed to the welcoming remarks. We respectfully invite Dr. Yeni Saryasi, SP MSc, Head of the International Seminar on Plant Protection 2026 Organized Committee, to deliver her welcoming remarks. Dr. Yeni, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning and warm greeting to everyone. The Honorable Guest, Director of University of Bengkulu, represented by the Vice Rector for Planning and Corporation, Professor Azhar Muda Lubis, the Secretary of the Indonesian Entomological Society, Dr. Maisarawati, the Secretary General of the Indonesian Phytopathological Society, Professor Ani Widyastuti, the Dean and Vice Dean of Faculty of Agriculture, the head of Department of Plant Protection, our distinguished keynote speakers, the dedicated organizing committee, and all participants and presenters join us via Zoom. First of all, let us express our gratitude to Almighty God for His blessing and grace, which allow us to gather today for the second International Seminar on Plant Protection, or ISPP 2026. Ladies and gentlemen, this is the second international seminar organized by the Plant Protection Study Program, University of Bengkulu. We are deeply honored to welcome 5 distinguished keynote speakers today. Dr. Cheng Fang Hong from National Chung Ching University, Taiwan, Professor Amelia Nicholas from Central Bicol State University of Agriculture, Philippines, Dr. Sofana Ramon from National University of Battambang, Cambodia, Dr. Norida Mazlan from University Putra, Malaysia, and Professor Agustin Zarkani from University of Bengkulu. We are also proud to welcome 47 presenters representing 15 university and academic institutions from Indonesia. I would like to express my appreciation to our dedicated organizing committee, university leaders, keynote speakers, presenter, participants, and all supporters who have made this seminar possible. I hope this seminar will be an inspiring for all of us. Thank you very much, and I wish you all successful and inspiring seminar. Enjoy the seminar. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Yeni Sari Asi, SPMSc, for your welcoming remarks and introduction to today's seminar.
We will now proceed with the welcoming speech by the Secretary General of Indonesian Phytopathological Society. We respectfully invite Professor Ani Widyastuti to deliver her welcoming speech. Professor Ani Widyastuti, the time is yours.
We have technical problem, so we will continue the next.
Hello, Tess. Is my voice clear?
Yes.
Yeah.
Ayo, silakan.
Okay, thank you very much. Distinguished Rector of Universitas Bengkulu, represented by Vice Rector for Planning and Cooperation, Professor Azhar Mulia Lubis, Distinguished Dean and Vice Dean of the Faculty of Agriculture, Universitas Bengkulu, Distinguished Secretary of Indonesian Entomological Society, distinguished colleagues and members of the organizing committee, honorable speakers and invited guests, researchers, academics, practitioners, students, and participants from Indonesia and abroad. Ladies and gentlemen, good morning. It is a great honor for me as the General Secretary of the Indonesian Phytopathological Society to contribute in this second international seminar of plant protection hosted by Universitas Bengkulu with the important theme horticultural plant pests and diseases. First, I would like to express my sincere appreciation to Universitas Bengkulu and the organizing committee for bringing together experts, researchers, practitioners, and students to discuss one of the important challenges facing modern agriculture. Ladies and gentlemen, horticultural crops are essential for food security, nutrition, farmers' livelihoods, and agricultural economies. However, their production continues to face serious threats from plant pests and diseases. Today, these challenges are becoming increasingly complex due to climate change, globalization, changes in cropping system, emerging pathogens, invasive pests, and the increasing resistance of pests and pathogens to pesticides. Climate change is particularly important because changes in temperature, rainfall, humidity, and extreme weather events can influence pathogen development, pest population, disease distribution, and host susceptibility. Therefore, plant protection today cannot depend only on conventional disease control. We need to move from a reactive approach to a proactive and preventive approach, from controlling disease after symptoms appear towards surveillance, early detection, risk prediction, and integrated management. This is where modern phytopathology has an increasingly important role. Advance in molecular diagnostic, genomics, metabolomics, microbiome research, remote sensing, artificial intelligence, and digital agriculture provide new opportunities to detect pathogen earlier, understanding plant-pathogen interaction, predict disease outbreaks, and develop more precise and sustainable management strategies. However, technology alone is not enough. We need to connect science with practice, laboratories with fields, researchers with farmers, and universities with industry and government. This is also our spirit in the Indonesian Phytopathological Society. We are committed to strengthening collaboration among academics, researchers, government institutions, industry, practitioners, and farming communities. International scientific forums such as this seminar are very important because they provide opportunity to exchange knowledge, establish new partnerships, and develop collaborative solutions to emerging plant health challenges. Therefore, let us use this seminar to move from diagnosis to prediction, from control to prevention, and from individual efforts to collaborative solution. Let us work together to develop plant protection strategies that are scientifically sound, economically feasible, environmentally sustainable, and beneficial to farmers. Once again, I sincerely congratulate Universitas Bengkulu and the organizing committee for conducting this important seminar. I hope that this meeting will generate new scientific insights, strengthen national and international collaboration, inspire young scientists, and contribute to a healthier and more sustainable future for horticultural production. I wish you a productive and inspiring seminar. Thank you very much.
Thank you, Bu Ani.
Thank you, Professor Ani Widi Astuti, for the meaningful and insightful remarks. Next, we invite the Vice Rector for Planning and Cooperation, Professor Asar Muda Lubis, to deliver the opening remarks and officially open the second International Seminar on Plant Protection 2026. Professor Asar, the time is yours.
Thank you. Honorable Dean of Faculty of Agriculture of University of Bengkulu, Prof. Suharyanto, and also, Vice Dean, I think it's already Prof. Heri, Pak Mustafa, as well as Bu Hesti. Our distinguished guests, Prof. Arrozi is still here. And then Dr. Henry Bustaman. Thank you for coming. Our respected keynote speaker, Prof. Chen from Taiwan, and then Bu Norida from Malaysia, Prof. Amelia from Philippines, Dr. Sapana from Cambodia, and Prof. Agustin from Indonesia as well. Invited speaker, researcher, academics, practitioner, student, and all participants of the second international seminar on plant protection, ISPP 2026. Assalamualaikum warahmatullahi wabarakatuh. First of all, I'd like to express our gratitude to Almighty God for His blessing today. So we enable to gathering together in this very important international seminar or conference. It is also my great honor, pleasure for me to welcome all of you to this very beautiful second international seminar plant protection with important themes, horticultural plant, pet, and disease. Ladies and gentlemen, on behalf of Rector University of Bengkulu, we'd like to say that this seminar is very important. Horticultural crop display vital role in supporting food security, nutrition, economic development, and farmer around the world. However, right now, as we can see that productivity and quality agriculture crop continue to face serious challenges from plant, pests, and disease. Therefore, we need integrated, innovative, environmental friendly, needs collaboration between scientists, science based on strategy that combine science in pathology, plant pathology, biotechnology, as well as remote sensing and artificial intelligence for sustainable agriculture. I think this seminar will provide an important platform to share your knowledge and then to share your exchange experience and then present your current finding. I hope also we can also develop collaboration between among countries like from Taiwan, Philippines, Indonesia, Cambodia, and Malaysia. I hope this scientific discussion during ISPP 2026 will not only contribute to advancement of knowledge, but also to practical solution. That's very important for farmer, stakeholder, to achieve productivity, silent, and sustainable horticultural system. I would like also to express my sincere appreciation to keynote speaker, invited speaker, reviewer, participant, especially to organizing committee who already managed this very wonderful meeting. Pak Mustafa, we can mark it as one of KPI, key performance indicator of Faculty of Agriculture, University of Bengkulu. And finally, I hope that ISPP 2026 2026 will strengthen our collaboration, our scientific network, establish new opportunity for research collaboration, both national and international level. And on behalf of Rector of University of Bengkulu, with great pleasure, by saying Bismillahirrahmanirrahim, I hereby declare that the second international seminar on plant protection ISPP 2026 is officially open. May this seminar be productive, inspiring, beneficial for advancement of plant protection and sustainable horticulture, particularly in Indonesia. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. WR4.
Sama-sama, Pak.
Thank you, Mr. Lubis.
Next agenda.
Thank you, Professor Azhar Lubis, for your opening remarks and for officially opening the second International Seminar on Plant Protection 2026.
Ladies and gentlemen, let us take a moment to pray for the success of today's event. We invite Revindra Harvis Wiryamanta to lead the prayer.
Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
Bismillahirrahmanirrahim. Assalatu wassalamu ala asrofil anbiya wal mursalin wa ala alihi wa ashabi ajma'in. Ya Allah, the most gracious, the most merciful, all praise is due to you alone, who has granted us life and the blessing of fertile land, growing plants, and abundant harvest. On this occasion, we gather to seek knowledge, exchange ideas, and strengthen our efforts in protecting, nurturing, and preserving your creation, particularly in the field of plant protection. Ya Allah, bless this international seminar. Make the knowledge we gain here a valuable deed that benefits all of humanity and the universe. Guide us to use the natural resources you have entrusted to us wisely and responsibly. Strengthen our resolve to maintain environmental balance, protect biodiversity, and sustain life on this earth. Ya Allah, the most wise, open our hearts and minds so that we always thirst for the knowledge you have bestowed. Let this gathering be a means for us to share knowledge and experiences, achieve better plant protection, benefit the nation, and make a lasting charitable act for all of us. Ya Allah, grant us strength and sincerity in fulfilling this responsibility. Protect us from negligence and excess in our treatment of the earth and your creations. Keep us away from actions that will harm future generations. Ease our steps as we carry out every duty you have entrusted to us, and pour blessings upon all our efforts. Ya Allah, the all-hearing, grant our prayers and hopes. Make this seminar beneficial and full of blessings. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar. Walhamdulillahi rabbil alamin. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Lip, Pak Dekan,
Ya,
Pak Henry, Prof.
ya, thank you so much,
Pak
Pak
Yozi,
Pak.
Lesti,
Okay.
Prof. Cheng, thank you.
Thank you.
Okay, thank you so much, Pak WR4.
Thank you. Before we begin the main session, We will proceed with the photo session. We invite our guests, keynote speaker, committee members, and participants to turn on their cameras if possible. The committee will guide the documentation.
Okay, ready? 1, 2, 3. Slide 2, ready? 1, 2, 3. Ladies and gentlemen, we will now begin the first keynote session featuring presentation by Dr. Cheng Fang Hong and Dr. Saufana Ramon, followed by a discussion session.
We respectfully invite the first moderator, Mr. Derry Gustian, SP MSc, to lead session 1. Mr. Derry Gustian. Mr. Derry Gustian is a lecturer at the University of Bengkulu, Indonesia. His expertise is in plant virology and molecular plant pathology, with research focusing on begomovirus, virus transmission, molecular detection, and virus-virus interaction. Mr. Derry Gustian, SP MSc, the screen and time are yours. For the first moderator, Mr. Dery Gustian is a lecturer at the University of Bengkulu, Indonesia. His expertise is in plant virology and molecular plant pathology, with research focusing on begomovirus, virus transmission, molecular detection, and virus-virus interaction. Mr. Dery Gustian, the time and screen are yours.
Okay, thank you so much for your introductions. Hello everyone. Assalamualaikum warahmatullahi wabarakatuh. My name is Terry Gustian. I'm one of the lecturer in the plant in the plant protections in University of Bengkulu. So, but now I'm studying in the PhD in the National Chongqing University in Taiwan. So now I will try to guide our first sessions. The first one I will I will introduce our speaker, because in the first session we have two speakers. The first one is Professor Hong Cheng Pang. Actually, Professor Hong Cheng Pang is one of my teacher in this school, right? So I know him. He is expert in the plant pathology, especially in the fungus disease. So he has many experience in the field, in the for the pest control and others. I mean, so I think it's time for you to ask question to him also. So, and also the other speaker we also have from the Cambodia. From the Cambodia, I will introduce also from the Cambodia. The speaker is Dr. Sopanara Mons. So, but the time for the stop is about 40 minutes. So each person will have like 20 minutes. So for the first speaker, I will let Professor Hong Cheng Pang from National Chongqing University. So for this talk, I will actually his expertise is in the plant pathology, particularly fungal and omnicepes disease. So his research focus on the plant disease diagnosis, like epidemiology, integrated disease management, and disease and several important diseases. So for the today the title is about Neostellidium dimidiatum, and emerging fungal pathogen affecting the dragon fruit and other crops. So now, Professor Hong Cheng Pang, I will give the time for you to give the presentation now. Please, Professor.
Yes. Thank you, Terry, for the introduction. I would like to share my screen now. And,
yes, Professor.
Yeah. So let me see. I'll share my screen. One. Can you see my screen?
Yes, Professor. We can see your screen now.
Oh, okay. Good. All right. Where is the? All right. I'll use the cursor here. Good morning, everyone. So I'm very excited and feel honored to be invited here to give the talk on the topic that we have been working in recent years. As Gary just talked, that I'm more focusing on the epidemiology and population genetics of fungal and all mycetes pathogens. So the topic for today will be Neostellidium dimidiatum, which is an emerging fungal pathogen on dragon fruit and also other crops. So my name is Zhenfang Hong. Yep. As we know, Neostellidium dimidiatum is an emerging plant pathogen And in recent years, it has been reported causing the shoot blight or the gammosis or internal browning or some fruit rot or the diebacks of in many plant hosts. And as you can see here, this is an almond causing the disease on almond, the disease on fig trees, and sometimes you will see the pycnidia on the twigs. And it can also cause the stem or trunk disease on grape or on the tomatoes. And lower right, you can see the fungal pathogen here. This is the other spores of the pathogen. But today we'll be focusing on the dragon fruit disease. So Neostridium demediatum can cause the disease called stem canker on dragon fruit. So initially you will see the clado. There are some tiny yellow sunken lesions on the clados or the stems of dragon fruit. These yellow sunken lesions will expand and emerge, and then they will coalesce and rot in the whole clado. This will eventually cause the yield loss or reduce the quality. The pathogen can also infect the fruits. So you can see on the right, the infected fruits were there are necrosis on the skins of the fruits. So this can cause a lot of loss and loss of quality or yield in dragon fruit industry worldwide. Not only infecting just plants, the pathogen has also been reported causing infections on humans. So this pathogen has been reported in 37 countries as an opportunistic pathogen that can cause onychomycosis, which is the infection of the toenails or the skin infections. And in some very rare cases, it can also cause advanced infections in humans. So this pathogen is not only just a human pathogen, but also can cause the opportunistic infection in humans. Then this also raised an issue of One Health. So when I was a postdoc at the University of Florida, I started the program, and when I come back to Taiwan, I started the research programs focusing on the epidemiology aspect and also the population genetics aspect of the pathogen. So because we already know the disease since 2012, but we have very limited information in terms of what's the conducive environment for the disease, and also what are the cultivars more susceptible or the phylogeny of the pathogen, etc. And based on the recent epidemiological studies, we also developed a disease forecasting system for dragon fruit stem canker. In terms of the population genetics studies, this is the part that I will be sharing with you today. So let's start with this. Assuming that we have a pathogen population, we cannot distinguish them just based on their morphology. But if we can use some molecular markers like RAPD, SSR, or single nucleotide polymorphism, we can distinguish them. So we can distinguish the pathogen populations based on the molecular markers or based on the sequences. Then we can investigate the genetic diversity of these populations. Then we can also investigate the mode of reproduction of these pathogens. For example, on the left and the right, these are clonal reproduced populations because they have the same genotypic characteristics. But if they go through, if the population go through recombination. We can see the sign of potential recombination here. And if we study that the population with some molecular markers or the sequences, we can investigate the migration and the genetic drift of the pathogen populations. And furthermore, we can even study the population differentiation associated with their respect hosts. So all these information will give us a deep insight into the ecology and the evolution history of the pathogen populations. With this being said, we started to collect samples in Taiwan. So we split Taiwan into 4 different regions based on the Central Mountain Range and the Zhoushui River. So on the right, this is the eastern part of Taiwan, and the northwestern part, southwestern part, and then the Penghu archipelago. We divided Taiwan into the 4 regions, and we collected the samples between 2011 and 2024 in these 4 regions. Of course, we also collected some of the isolates from the isolates were shared by our collaborators in different research institutes. And in addition, we purchased some reference isolates. There are 8 non-Taiwan isolates were purchased from the Westdike Fungal Biodiversity Institute. So we have more than 200 isolates in this study. So firstly, we used 3 housekeeping genes to distinguish the population. So we use the ITS, the TEF1 alpha, and the beta tubulin for identifying the populations. So in this part, let's focus on the shadow. The green shadow means those are the sequences from dragon fruit. And some of the sequences were obtained from the NCBI gene bank. And the dark black ones are the isolates from Taiwan. So if we just look at the ITS regions, you can see the majority of the dragon fruit isolates were clustered together. And the red dots indicate the 8 non-Taiwan isolates from different hosts. And something interesting is that the dragon fruit isolates from Puerto Rico is somewhat different. So that means in a larger or global scale, there might be some diversity from dragon fruit. So if we add some more housekeeping genes, we add the ITS and the TF1 alpha, we can see the dragon fruit population still clustered together, and the Puerto Rico is still here, and the purple one is the population from Catalea orchids. If we use 3 housekeeping genes, we still see the majority of the isolates from dragon fruits cluster together, no matter the geographic origins. And here is the population from Catalea orchids, and the rest, the red dot, indicate the isolates from different hosts. These are the phylogenetic analysis. But if we look at the phylogenetic network, this also indicates there is a limited genetic diversity in the populations from Catalea orchid and from dragon fruit. But at the global scale, you can see there is a higher diversity in the Neoceledium demediated population. So it looks like there are some association or differentiation associated with the host. But this has to be very cautious because we have unbalanced isolates from different hosts. So in the future, we can do further collection from different hosts. And here you can see the dragon fruit isolates were clustered together, and the Catalea orchids were isolates were clustered together. So in order to further investigate the diversity, we can see at the global scale there is a higher diversity in the Neoceledium immediatum population. And in Taiwan, the population in Taiwan has a relatively low genetic diversity. And something interesting here is that the RM value that indicates potentially the cryptic recombination in the evolution history of the pathogen. So although the sexual reproduction has not been observed or reported in this pathogen, but in history, there might be some recombination event occurred in the history. To further study the spatial temporal genetic structure of the pathogen, we developed 11 simple sequence repeat markers. And as you can see here, the population in red indicates the populations are from non-Taiwan population. And the green indicates the population from Taiwan on the Catalea orchids. And the blue ones indicate the populations from dragon fruit in Taiwan. So for the dragon fruit and Catalea orchid populations, you can see most of the loci were fixed, but in the non-Taiwan population, only a few loci were fixed, and there are higher diversity in the allele in different loci. So the 11 simple sequence repeat markers is sufficient to distinguish all the multilocus genotypes that we studied in these 244 isolates. And in total, we identified 13 multilocus genotypes, and I'll call them MLGs hereafter. So there are 8 different MLGs from the 8 non-Taiwan isolates. And in Taiwan, we only found 4 MLGs from dragon fruit. And from the Catalea orchid, we found only 1 multilocus genotype. And here is a genotypic diversity based on rarefied samples. So this table, this figure tell us that there is a higher genotypic diversity in the non-Taiwan population if we compare them with the same sample size, and there is a very low genotypic diversity in the dragon fruit population from Taiwan, as well as the population from Catalea orchids. If we analyze the populations based on their geographic origin, we can see here each circle represents 1 multilocus genotype, and the size of the circle is proportional to the number of isolates. So from the dragon fruit population, there are only 4 MLGs we found, and 1 clonal lineage is dominating the population in Taiwan. And for the Catalea orchid, all of them belong to the same multilocus genotype. And this part is the rest of the isolates that are non-Taiwan isolates. So if we further analyze the pathogen population based on the collecting time, we can see from 2011 to 2024, majority of the isolates are belonging to the same the same multilocus genotype. That suggests the Taiwan population is basically predominantly a clonal lineage is affecting the dragon fruit in Taiwan, and only 3 different multilocus genotypes could be found in the affecting dragon fruit in Taiwan. So to further investigate the population differentiation, we compared the non-Taiwan isolates with the dragon fruit isolates and the Catalea population. So we found there are higher differentiation among these populations. But and if we compare the Catalea population with the dragon fruit population, they are also quite different. But among the populations, the dragon fruit populations from different locations, they are very similar. So that indicates there are some potential association with the host. I mean, the population differentiation may be associated with the host, but still we need to be cautious with this conclusion because we have unbalanced sample size in dragon fruit and Catalea orchid and other and the isolates from other hosts. So this is a PCOA analysis. This also shows that dragon fruit population clustered together and the non-Taiwan isolates were scattered here. And the Catalea orchid seems to be closer related with these different isolates. So for this part, we found a higher genetic diversity in the population from various hosts and the geographic origins at a global scale. That may suggest, and some of the information may suggest a cryptic recombination in the evolution history in Neocellulian dimidiatum. And we found a low genotypic diversity in Neocellulian dimidiatum populations in Taiwan. That may suggest the population has been affected by the boundary effect or the genetic drift. And a single clonal lineage is dominating the population in dragon fruit, so in Taiwan. So in the future, because we have some evidence from other, based on the sequences from other countries, but we don't have sufficient isolates, and there are some missing data from the NCBI gene bank, so we cannot really compare the population in a larger in a global at a global scale, but at least in a smaller scale in Taiwan, a single clonal lineage is dominating on dragon fruit and causing the problems in dragon fruit. And they are the population is sustaining via the clonal reproduction. So this is the primary mode of reproduction of the pathogen in Taiwan. So to further investigate the phenotypic diversity, we ask some questions about are there any variations in the morphology or in the their fitness traits or their pathogenicity or their latent period? Because we already know that there is a very limited genetic diversity, but how about the phenotypic diversity? So we started to look at the morphology, and there is a very limited variations in their morphology. But when we look at their fitness, the effect of temperature on the mycelial growth, there are some somewhat variations based on the locations. I mean, in some regions, the population seems more adapted to higher temperature, and compare with some regions, they are not as adapted to the environment. So there are still some variation in their phenotype. And also the isolates, when we inoculated them on the cladles, these green parts are the isolates collected from dragon fruit. You can see it seems like to cause more severe diseases. And the purple parts are the isolates collected from the Catalina orchid, and the rest are collected. These parts are the isolates from other hosts, and these are non-Taiwan isolates. But all of them are pathogenic to dragon fruit. And on the lower right, this is the control. So it seems like there are some variations in the pathogenicity, but when we do the statistical analysis, they are not statistically significant. That's probably because the variations among clados, because they are not really showing a consistent lesion. But the latent period has something interesting, because if we inoculate those isolates on the clados, some of them will show will start to sporulate, or they will produce the secondary inoculum significantly earlier than the rest, the non-dragon fruit isolates. So these results suggest there might be host-associated adaptation in these clonal populations. So for this part, we observe no morphological variations among isolates, but there is a significant differences in the hyphal growth and the spore germination rates in the clonal population. And that may suggest the adaptation in different regions of the pathogen in different regions. And there is a significant variation in the latent period, and that may suggest the host-associated adaptation, along with the evidence from the genetic diversity. We suspect that this pathogen is adapting to their specific host, but this has to be cautious, because we don't have equally number of isolates from different hosts. So in the future, we may have some collaborations, or we may collect more samples from different hosts, trying to investigate their genetic diversity at a global scale. And we can study if the clonal expansion is a real thing at a global scale, because in Taiwan we already see that clonal expansion, maybe along with the expansion of planting materials, and this pathogen seems to adapt at a different region. So with that, I think that's my talk, and I would like to take any questions. And I would like to thank all the funding agency and collaborators, and also my students in the lab. So thank you.
Thank you so much for Professor Cheng Pang Hong. Okay, Professor Hong. So for the discussion, we will have discussion later after the second talk from the Dr. Sopanara Mon. So hold there first. Okay. Now we will move to the second speaker from the first sessions, from Dr. Sopanara Mon. So Dr. Sopanara Mon is from National University of Battambang, Cambodia. So his expertise is in agronomy, crop productions, and agricultural extensions. His research focus on particularly on cassava propagations, plant growth regulators, aeroponic production system, and sustainable crop productions. So the title of the presentation today from him is like crop pests and disease control for sustainable agriculture. So now, Dr. Sopanara Mohon, please.
Yes, yeah, thank you so much for your coordinate. Please, you share the slide presentation for me because my computer now is error. That's why I send my material in the.
Okay.
Yeah, thank you so much.
Okay.
Please. Sorry, my computer today is.
It's okay.
Yeah.
So now, Dr. Geni Saria, she still share the.
Yes, thank you so much. For my topic today is just the kind one, not the like the sustainable agriculture from the plantation. It's just my case study from my research in aeroponics. I will show you. Yeah.
I think it's very good talk. I think it's very good, and we would like to hear from you directly about your experience.
Yeah. Thank you so much.
Yes.
Okay.
Now, where is the slide? Please.
Yeah.
Okay. Now. Okay.
Thank you so much. Yeah. Okay. My name is Sawan Ramun. I'm from National University of Battambang, Cambodia. Maybe some of you know about Cambodia or the National University of Battambang. And I graduated from Thailand, from Ubon Ratchathani, and here project. So today I would like to share about my research on the topic development of combination rapid propagation technique for diverse cassava cultivar in aerobic system. Next. As we know that the cassava production in the world is like our Asian, like Indonesia, Thailand, Cambodia, Vietnam are the top 10 of the cassava production in the world. Next. And during the cassava is more important or have the many advantage of the like to produce the oil or the organics and the other like to use the waste and also use the fertilizer and animal feed. Next. But even though there are many potential or the potential and also the benefit for cassava, but it is facing the larger we call cassava mosaic virus that caused by virus, and its yield loss and from 20 to 90% up to the infected infection status. It is very dangerous. That it's you know that it's the virus it couldn't treat, and it is happen that we face this challenge. To address this challenge, we don't have the any tricks, so we just try to define the method to propagate the herbicide, use the herbicide for the address the challenge. Like we have like the conventional practice, like cut the stem around 20 centimeter, and for the other research they find to the mini cutting like 5 centimeter meter, and also the other research also use the leaf bud. They just cut the one leaf bud from the stem. Next. And also the other research try to find another way to propagate cassava production, like use the hormone and also the aeroponic system to propagate this cassava. And in this case, when I found that there is no like the gap that we don't have to use the IBA with the lipat. So I just try to propose this topic, and also I found that when the previous research they use the greenhouse with condition, temperature, and lighting for the propagate the cassava. So I would like to investigate that is work or not with the simple greenhouse. Next. So I propose the three main three sub experiment. The first I testing media on the list bud cutting, and the second I compare the three propagation method to propagate the cassava, and the last experiment is the propagation under aerobic system. Next. So the first experiment, I try to adapt the previous research to cut the leaf cutting. I think maybe all of you maybe it's like a never see or let's see to see this for the just a small leaf cutting, and use the aeroplane with we have a factorial experiment like the media and also the variety, and we have collect some many red. We use the IPA IBA 250 mg, and also the SAS, and under the conditional simple greenhouse for 40 days. Next. For the second experiment, I propagate the three techniques like I want to compare the leaf bud cutting and mini cutting and normal cutting to how they perform the develop or the propagation, and with the collect all some thread for duration 2 months. Next. And for the final step, the last experiment, I conduct the bacterial experiment that use the leaf like the new suit from all experiment to extend to more propagate under aerobic system. The first I prepare the media solution, and I cut and separate it by and use the under the simple greenhouse to observe that it is possible or not if we don't have the light and the air conditioning for the control the environment. Next. And I use statistic 10 and R program to analyze my data. Next. Yes, as the result for the first experiment, like the I can call a sub experiment, the we have the two factor and also factorial. The first the from the KU cultivar compared to the Rajong 9 cultivar compared to KU 50 and Hoibong 60, it's better grow from the leaf bud or survival rate. It is more observed. And also for the distilled water for the and the SAS solution is also much better than IBA and SAS nutrient. In this finding, I found that the young tissue is not suitable for the hormone IBA, even though we just use the low concentrate.
Apa tuh sih lagi MPJP, SPKPGA 3, JAGI PRB, TGSR.
As next, yeah. And for the principal component analysis, we found that the most of the thread is better in the more of the thread is better in the Prajongnang, and also explain most dimension also, and for the media it's found that the SAS and the control distributor is much better performance either from the data set for the biflot. Next.
Okay, next.
Sorry, image.
Oh, please, next, please. Hello, next. Okay, so you can continue.
Yes, for the we found that the aeroplanes like the media is not suitable for the IBA. That should I recommend it. And for the next experiment is the compare the three propagation. The next is the result.
In this slide or in other slide?
No.
Which slide? Doctor?
Yeah.
Okay, this one. Okay.
No, one more. Experiment 2, the result in experiment 2.
The result of experiment is
experiment 2.
Experiment 2.
I think.
Oh, this
yeah,
one.
yeah,
Okay.
yeah.
Okay,
Yes.
okay, this one. Okay. Sorry.
Yeah, 13, yeah, okay.
You can continue.
Okay, yes. And for the this compare for the three propagation method, we found that the survival rate is much better in mini cutting, and for the like flank row development is and propagation is better for the normal cutting for the 20 centimeter. Next. And it means the biflot for the data set is found that the most growth and development is better or greater than in the normal cutting, but the survival rate is better in the leaf cutting. Next. Yes, and this is the result from the observation in the under simple greenhouse. That in my gap, I found that it is possible or not to propagate under just a simple greenhouse, don't have the air conditioning and also the light. It is possible to it's possible to be it. I found that is much higher survival rate and also net. Yes, as the result for the cultivar, it is not different for all thread, but for the leaf bud and mini cutting is survival rate is much better than the normal cutting. During observation, I found that normal cutting is look like the tissue is younger and some face like fungi as well. Next. And also for the beef lot PCA, also the explain much high the percentage, and also the most survival rate is should be used for the leaf wat and mini cutting. For the normal cutting is not suitable. Next. Yes, as my conclude is I found that the SARS and is the optimal mediate for propagation on the growth rate and survival rate, and for the cassava variety like Rajong 9 and 560 is also better, and aeroponic is also the another method to propagate cassava or other plant to reduce the time if we don't want to like for the maybe for the key farmer, they don't have like the tissue culture, they just want to use the simple way. Yeah. Next. And for the study found that it is possible to use the aeroponic, and maybe we can select the other method or suitable method that you have in the real situation. Maybe you can use the mini cutting or leaf cutting for the propagate. Yeah, that's all for my sharing. Yeah, thank you so much.
Okay, thank you so much for Dr. Sopana Ramon. I think the talk is very excellent because about cassava, right? In Indonesia, we also have a lot of cassava. So now we are good for the discussion sessions. I think for the Dr. Hong, Professor Hong, are you there?
Yeah, I'm here.
Yeah, okay, Professor. In the comment in the chat box, we have some question for you.
I read it.
The first question is from Mr. Leonardo Silalahi. The question is, given the distinct hyper growth response to different temperature, is it possible to use this data to develop the climate-based disease forecasting model to help growers predict and prevent the outbreaks before the lesion occur? Okay, Professor Hans.
Yes. Can I share my screen again?
Yes,
And
yes, of course.
I can show some of the results.
Yes.
Let me share my screen again here. So can everyone see this? Okay.
Yes, Professor.
Okay. So this part of our result that we use the information obtained from the mycelial growth and the spore germination. We have built a simulation model that can predict the risk of infection based on the temperature and the humidity. And of course, this is an earlier version of the prediction model for dragonfly stem canker. We have a later model that I didn't show here, but basically this is the information that we can use the temperature and the humidity and the latent period or the incubation period to establish a prediction model. And we are currently working on this now, and we have we have already established some prediction models for dragonfly stem canker, and we are currently also validating the model in Taiwan. But if anyone would like to collaborate or to use the model, you can just inform us. Maybe, yeah. So So basically, yes, we can use the information for establishing the prediction model, and that's what we want. What we want to do to prevent or predict the risk of the disease, and then we can take the preventive strategies beforehand before we see the lesions. And so that's the first question. Hope that will answer your question.
Okay. How about Mr. Leonardo? Can you respond that? How about, did you okay for the answer? Mr. Leonardo? Okay. Mr. Leonardo, are you there? Or if not, we go to the second questions now. For the second question is also to Professor Hong, is from the Mrs. Nurlianti Gayer Pangidu. The question is, what are genetic drift and the founder effect, and what is their significance in the bioecology and significance in the bioecology and control of Neolithelium dimidiatum? And the second question is, based on the conclusion of your research, is there a specific mechanism that enables this pathogenic fungus to infect both plants and humans? Okay, Professor Hong, please.
Yeah. So the genetic drift and the founder effect, they are very similar concepts, but a little bit different. So for the genetic drift, it means a random loss of some genotypes in the population. When the population, when the pathogen population, you would assume in the source of origin, they will have a higher diversity. But when a small colony or a small part of the population start to colonize other hosts or go through or carried by human to other locations, there would be some random loss of some genotypes. So that is genetic drift. And the founder effect is based on the genetic drift. So the founder effect is that when the population comes to a new location, for example, like at the global scale, the Neosteridium dimidiatum has a higher diversity, but when it comes to different locations or at different hosts, the pathogen has to adapt to the new host. Otherwise, they will not be able to survive. So in the early generations, we call them the founder. They may have some loss of the genetic diversity. So that's a founder effect that a new population may face when they are moving towards a new locations or new hosts. So in terms of the infection to plants and the humans, we don't know yet clearly about the mechanism. So there are several plant pathogens that could have the opportunity to infect both humans and plants. But for this one, I cannot tell you for now because we are still working on that. That's why we also have a new program that will study we will use comparative genomics approaches to study the genetic structure of or some compositions among different isolates that from humans or from plants, and then see what could be the mechanism. So for the second question, I cannot answer you for now. Yeah. Does that answer the questions?
Is it answer your questions, Mrs. Nurlianto? Miss Nurlianto Pangidu? Are you still here? Can you respond for the answer? Okay, Professor Hong, I think.
Yeah, we can move on. Yeah, I think we can move on.
Yeah, I think we can move to the third question from the Caca Handika. So he is a master student at the University of Bengkulu. So he want to ask, how can we ensure that the difference in genetic structure observed in the Neoscitaligium populations are truly attributable to the geographical factors rather than to the difference in host plants and environmental conditions or sampling bias? Please, Professor. Yes.
Yes. So this is a very good question and an interesting question because when we are doing this kind of research, usually we will collect a lot of samples from different locations or hosts and kind. So that's basically based on the questions you are asking. So we can only collect samples as possible as we can. So for example, like if I would like to ask the question about the evolution history of the pathogen, then I have to collect the samples from different years, and as as maybe I have to collect the very similar number of isolates in each year. But sometimes, it's quite different. I mean, it's quite difficult because we may not have the opportunity to collect those samples. So sometimes it could be a little bit biased. But if we have sufficient number of isolates, like usually we would say more than 30 is a generally okay number, but it would be better if you have more than 100 isolates from different locations, from different time, from different hosts. That will reveal somewhat the genetic structure of the pathogen, and without, I mean, of course, it always comes with a bias because we cannot really collect these many isolates from either each locations or years or hosts. So that's the limitation. But we can do as much as we can to minimize the bias. So if we can do that, we have a good sampling strategies. Then the next is the issue of the genetic markers, the molecular markers, because some of the markers are good, some of the markers are not as good because they will be affected by selection or by the natural selection or by some other factors. So we also need to use the right molecular markers. So SSI is one of the most used markers because it's a codominant, it's a higher polymorphic, and it's assumed neutral. So so those are the points that when we are doing the research, we have to think beforehand. Yeah, I hope that can answer your question, and hope that you if you are interested, then you can start read some of the books or papers associated with that. Yeah, that's it.
Dr. Handika, I see you are there. Maybe you can talk little about the questions, about the answer. You want to talk something? Or? No?
Right, so I cannot hear you. You mute yourself.
Okay, Professor. So for the next question is also for the Professor Hong. The question is about the how can one distinguish between a plant system caused by bacterial, virus, and animal infections? Thank you.
Uh, for the plant, the symptoms on the plant, caused by bacteria, virus, or fungal pathogens, they sometimes they will be very similar, but there are some very distinct symptoms caused by certain pathogens. For example, for the viruses, usually they will cause mottling or mosaic or distortion of the leaves, and uh, but for and then sometimes the necrosis symptoms. But these symptoms could be confused with the fungal infections or bacterial infections. And for the fungal pathogens, they can cause swelling, gout, necrosis, or chlorosis, some lesions, fruit rot, etc. And for the bacteria, usually they will cause the symptoms with the yellow halo or necrosis, and some rotting, macerating of the tissues. So it's if you are just distinguishing the diseases based on the symptoms, it could be difficult because some of the symptoms will be very similar. But when we are diagnosing a disease, usually we will, based on the information such as the host, the cultivar of the host, and the cultural practices, and we will, of course, we will also look at the symptoms, and we will look at the signs. The signs are the, 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 the pathogen itself from the symptoms. So if we still cannot distinguish them, then we will use cultural media, or we will use some molecular tools, and there are several tools. So it's not easy just distinguish it's not easy to distinguish a disease just based on the symptom, because there are many diseases that could be confusing based on symptoms. So in terms of the animal infection, well, that's not my expertise, but for the plants, I can tell you that we can use this many different strategies to diagnose the diseases. Yes, that's a easy answer for this. Yeah.
Okay. Thank you so much, Professor Hong. And we have the last question for you here from the Ulfa. So she asked about what molecular markers do researchers use to distinguish pathogen populations and investigate genetic diversity? Okay, Professor.
Okay, so there are several molecular markers that we can use based on their, I would say, resolution. And also, as I mentioned in the earlier time, when the sequencing is not so easy, so we can use the AFLP, RAPD, and SSR, etc. Those are the molecular markers that we can use, and some would use the enzymes. And for now, we have sequencing technology, so we can use the Sanger sequencing. We can use the next generation and third generation sequencing for finding the single nucleotide polymorphisms, and we can also do the whole genome comparison or some comparative genomics studies. Those are very useful tools that we can use to study the pathogen population, or to compare the genome of the very similar pathogens. So those are quite useful tools. Yeah.
Okay, Professor. Thank you so much for your answer. I think that's all for your questions for your presentations. And I would like to thanks for your presentation and for your great talk in this conference, I think it's very important for us. Maybe in the future we can make some collaborations between our university, right? That's it. Okay. Thank you so much, Professor. So, and now we will move to the question for the Dr. Sopanara. The first question for Dr. Sopanara is like from you, Teresa Cahyaningrum. The question is like, I will to ask if the RY9 performs better overall. Does this means RY9 is the best cultivar under all media conditions? And what are the suggestions for the future research? Okay.
Yes, thank you so much for the question. My observation and also research finding, I found that Rajong 9, we call Rajong 9, the abbreviation or Y9, Rajong 9 is better, but it's not much for the most media or the solution, because due to my observation, even though it's promote the number of the root and also the stronger, but it's still the survival rate is not like acceptable. I think when we use the hormone, we need to check carefully for the concentrate and how it's affect to the plant. Yeah, I can. Yeah.
Okay. How about you, Teresa Cahyaningrum? How about the answer? Maybe you can respond.
Yeah.
So she asked about the. I want to ask to Dr. Sopanara, what are the characteristics of the good cassava cultivar seeds? Please.
Yes, thank you so much. According to my finding and experience, in fact, in first, I never imagined that the leaf bud can like propagate. It is like the one thing that I surprised also when I found that it's also one kind of the propagation. Generally, we use just the 20 centimeter and 5 centimeter for the propagate. But when I found that this is I very interested, and also I try to adapt and develop another. And I found that it is the young seed is much better if we when I propagate is from the leaf bud and also from the mini cutting is the tissue is stronger than the shooting from the normal cutting. That's why it's a good performance under aerobic system. I would clarify that I use the nutrition in the aerobic system around only 10 days to promote the root, but it is also promote the root and get the high survival rate. After that, I transplanted to the soy pot with the same pot media like the just biochar and combined with the compost a little bit. Yeah, and for I think for the cassava seed is you need to take care about the leaf. If the leaf like simply green and the leaf is smoothly not curly and not infected by the red mice and whitefly is much better, because in my case I very be careful with the red mice that cause the yellow and also the white and it is very dangerous as well. Just 2 weeks they will make all the cassava seedling, and also the whitefly that you know that whitefly is the vector that can transmit the virus from one plant to one plant. That I think all of you may know about this. It is my my knowledge. As my knowledge, I can suggestion that when you select the cassava seedling, you need to take care about the under the leaf. You can find the red mire knot, and also the simple or the like the physiological thread or the green leaf. Yeah. Thank you.
Okay, thank you so much for Dr. Sopanara. How about that for Lydia Afriani? Did you okay with the answer? Okay.
Yeah, one more. Yeah.
Okay,
For
and
the,
huh?
yeah. Go ahead.
Oh, thank okay. Thank you, Dr. Supana. So, Dr. Mon. So, the next question is from Dr. Supanjani. So, he is also the professor in agronomy in University of Bengkulu. So, his question about the I'm sorry for missing the informations about the SAH solution. Could you explain this solution? Thank you.
Yes. Sorry, I didn't put the full like SAS nutrient, like we call semi SAS nutrient with semi automatic hydroponic. Yeah, maybe I will listen to this chart. Yeah, it's the one that we combine the all nutrition from. Yeah, maybe you can find in the my slide. I found in the material. You see the
Maybe you can share the slide, Dr. Sopanara, and show
yes, thank you. Yeah. There is the combine all the nutrient like the nitrogen, and also for the main like the sodium, and also the other that we combine together with the. And if you found my paper, you will see the real like the sales, like the real practice, the amount of the concentrate that you can adapt. And also I right now I also adapt this to other plant or to more propagation, because I want to know that beside the cassava, maybe the other that the plant that difficulty to propagate, maybe I just want to observe if it is work with the other, it might be good, because just promote the root is like a 10 day, but we depend on our target and also the propagation as well. Maybe I found that one guy that they mentioned about the coffee, maybe they want to to use this solution to propagate for the coffee as well.
Have you used that before, Dr. Man? Have you used the coffee solution or something before?
I just use for the cassava, and right now my student observe on the rows. Yeah, before we from the horticulture, and in fact I am the head department of agriculture and work in the horticulture, but my study is in agronomy. Right now I try to research in about on the horticulture right now. Yeah.
Okay. And next question from Fitri Yali. So she is from the University of Lampung. So her question is about, could I have your contact, such email or something other? If you don't mind, I would like to discuss about my cassava research. And may I have your presentation materials, PPT slides? Thank you.
Yes, welcome. Yeah, thank you very much for your nice interesting, and also want to collaborate. Yeah, I want to collaborate about my research as well. Right now I try to find the like make a cooperation with like the Asian university as well. I will I will send you and I will send to Dr. Henry. Maybe Dr. Henry can transfer my all information to him. Yeah. Thank you so much.
Thank you so much, Dr. Sopanara. I think that's all for the question for you for today. For today talk. I think your talk is very excellent today. I really got many information from your presentations. Thank you so much for that, Dr. Sopanara.
Yeah, thank you.
Okay, and now as a moderator for the first sessions, I would like thank you to you all guys, and I will give back to the committee, please, and we will go to the next session, session 2. I think Dr. Henry Bustamam will guide the discussions. Okay, thank you so much for the first session. Thank you.
Thank you, Dari Gustian SPMSC, for leading the first session. We also sincerely appreciate Dr. Cheng Fang Hong and Dr. Sofan Naramon for their insightful and valuable presentation.
Ladies and gentlemen, as a form of our sincere appreciation, we will now proceed to the symbolic certificate presentation for the keynote speaker and moderator.
The first certificate of appreciation is present to Dr. Cheng Fang Hong from National Chung Hsing University, Taiwan, as a keynote speaker. The second certificate of appreciation is present to Dr. Sofan Naramon from the National University of Battambang, Cambodia, as a keynote speaker. The next certificate of appreciation is present to our moderator, Dari Gustian, SPMSC, for his valuable contribution in guiding and moderating the discussion session.
Ininya nggak ditampilkan ya?
We express our highest appreciation to the keynote speaker and moderator for their meaningful contribution to the second international seminar on plant protection. Ladies and gentlemen, we will now continue with the second keynote session. We respectfully invite the second moderator, Dr. Ingenieur Henry Bustamam, MS, to lead session 2. Dr. Ingenieur Henry Bustamam is an associate professor at the University of Bengkulu, Indonesia. His expertise is in plant pathology and biological control, with research focusing on antagonic microorganisms and sustainable management of plant diseases.
The second session will be featured keynote presentation by Dr. Norida Mazlan from University Putra Malaysia, Professor Agustin Zarkani from the University of Bengkulu, Indonesia, and Professor Amelia Nicolas from Central Bicol State University of Agriculture, Philippines.
Dr. Ingenieur Henry Bustamam, MS. The screen and time are yours.
Okay, thank you for this session. Assalamualaikum warahmatullahi wabarakatuh. I'm Henry Bustamam from the University of Bengkulu. I would like to guide this session. We please, first, Professor Dr. Norida Mazlan. She is from University Putra Malaysia. Her expertise is entomology, pest management, and pesticide management. And her research focus on insect pest ecology, pesticide use and resistance, and sustainable pest management in agriculture system. Yeah, this session, she will present the paper with title, The Role of Nutrition in Determining the Three Topic Interaction Between Quebec Plant, Diamondback Moths, and its Parasitoid. Hello, Dr. Norida Maslan. Good morning.
Hello, Dr. Henry and
How are
everyone.
you?
I'm
Okay.
fine, thank you.
Okay. Second, Professor Agustin Jarkani. Yeah. He is from University of Bengkulu, Indonesia. His expertise is in plant entomology, insect pest management, and particular mealybug taxonomy. His research has contributed to the identification and description of mealybug species in Indonesia, as well as the development of mealybug identification resource and biodiversity database. Yeah. This session, he will present with title Mealybug on Horticultural Plant in Indonesia. Hello, Professor Agustin Zarkani.
Assalamualaikum Pak Henry, Bu Nur.
Yeah. How are you?
Fine, thank you, Pak Henry.
Everything nice, yeah. Nice, good looking, yeah, today.
Thank
okay?
you.
Okay, third is Professor Amelia R. Nicholas, yeah. She is from Central Bicol State University of Agriculture, the Philippines. Her expertise is entomology and stingless bee science, with research focusing on stingless bee taxonomy, meliponiculture, pollination, biodiversity conservation, and meliponitourism. She is also actively involved in international collaboration on stingless bee conservation and sustainable agriculture. This session, she will present with title Advancing One Health Through Pollinator Center Horticulture. Hello, Prof. Amelia Nicholas. Good
Hello,
morning.
Dr. Henry. Good morning. Good morning, everyone.
How are you?
I'm good. Thank you for inviting me to this
Okay. I think you
wonderful.
look so beautiful, yeah?
Thank you very much. Thank you.
Okay, I think we let its speaker can present the paper around 20 minutes, yeah, and then we continue with discussion. Okay, in the first time on this session, I let Professor Norida Maslan to present her paper. Please. Time for you.
Okay, thank you, Dr. Henry, for moderating these sessions. So I think I will share my slides for the presentations. Yeah. Okay.
Yes.
Hold on. Yeah.
Please, Dr. Yeni, please.
Is it allowed to share?
Okay.
Okay. Should I go for full screen?
Okay, yes.
But check for the this one. I have to go for the full screens. Sorry, can everyone see? Because this like is I can find the full screen here. This is comment. I cannot cover. Okay, let's see. Can everyone see? I still cannot get for the full screen. Sorry. Or maybe the organizer can go and share the screen then. Although I prefer to do it my own, but let's see.
Okay, Bu Yeni, please take the slide board. Yeah.
Yeah, I cannot find slide mode here. Oh, I have to stop sharing.
Sorry, Dr. Nafida, can you share the PPT in the chat?
I sent this morning. Did you receive it?
Not yet.
Not yet.
Yeah.
Okay, in that case, I can only do the halfway. I cannot share the full screen.
Oh, it's okay. No problem.
Is that okay?
Yeah.
Then I can do it, but I just can share the full screen. But I can't find the. Sorry, let me adjust here. So
There are slideshow on the top.
yeah.
Any animation?
Yeah, slideshow here. Oh, wait. It keep coming up.
Oh,
Yeah. I put slideshow. There is nothing here.
You can set up slideshow. Okay.
This auto.
Yep. Susah ya.
Still didn't go for the full slideshow. Okay. Okay, so now yeah. Okay, Mr. Assalamualaikum and good we stay in the morning. Good morning to the our moderator, Dr. Henry Bustaman, and all the speakers of the ISPP 2026. So I would like to thanks for the organizing committee for inviting me to present, sharing the sessions in this seminar. So today I would like to present the title of the role of nutrition, meaning the tritrophic interactions of between cabbage plants, DBM, and its parasitoid. So I'll say that talking about cabbage, I think this is quite an old study where we know the main pest for the cabbage, we have a dimobacterium. It's not only attacking cabbage, but most of the cruciferous plants. So, and why actually DBM, they are specific pests. So most of them attack the cruciferous plants, because means we have a very specific VOC which attracts the pest. So and Poticia protella, like we have like in Malaysia we have Diadema and Poticia. So like the Diadema was mostly known for the raw parasitisms of DBM in the highlands, but Poticia is on the lowland area. So like in my studies here, we are using the parasitoid Poticia, where is used to its importance of the parasitoid for the DBM, especially in the lowland area. So well, I would say talking about the DBM. So most of the time, the common practices of the pest control for the DBM for many years, for decades, people keep looking into the using the pesticide. So any problems is that you find the pest, they go and keep using pesticide, and the pesticide, the pest become resistant to the pesticide. So and they keep changing a different type of the active ingredient, and the same thing happen. But when we're talking but actually we're talking about pest management, it's not the way only taking pesticide is just like try to eliminate the solve the problem, the pest itself. But in the whole of the pest management, we have to look at the whole crop management practices. So like in I mean most of the time with when we found the use of nitrogens, where it can help to grow the plants, I mean increase the food productions. So everyone busy, say okay, go and we need lots of nitrogens, and because nitrogens in we put nitrogens, the plants will grow very fast. So it's become very important source of the agrochemicals in our country. So it because we also want to increase the the world population increase, we want to increase the food because of food security, and so therefore the needs for the nitrogens also increase. So then we're just talking about we keep using a lot of nitrogens, and actually how much when used, we apply it, actually this nitrogen actually only 50% used as a used in the plants, okay, for the grow you get, and because it's used into different types of the functions in the growth of the plant. Yeah, okay. So now we're talking about how nitrogen method assimilations, okay, with the high end fertilizations, of course it will increase the N, so therefore the ratios of the CNNs is low, is getting lower. And glucosinolates is very important, especially in the cruciferous plants, because it's actually is include the have other essential amino acids like methionine, tryptophan, phenazines, because this is needed for the GLS biosynthesis. And why the GSL is very important, because I found that it's also quite related to the it's actually is a VOC in the plants that release when the plant has been attacked by the predator, by the herbivore, and then also it will attract the predator. So GSL also is it have a sulfur, primary sulfur, and nitrogen rich secondary metabolite as a protecting plants, because this is the contents of the GSL actually is very important, because nitrogen contents in the plants will induce either because GSS is also sometimes is protect the plants where it has a big functions of to reduce the growth of the herbivore. And then this myrosinase actually is where it's released when the tissue damage, then it's release the damage, it will bring DSL into contact with the myrosinase, then forming the toxics. This is the toxic that is actually is play a functions of the plant's defense. Okay, so when talking about tritophic levels of the interactions plants pests and the natural enemies. When talking about, we have 3 levels. Okay, So the 3 levels here, we have started from the host plants. Level 2 is the herbivore itself, and then the third is a natural enemy. So basically, when talking about, it's all started from the nitrogen input, where it's directly will impact the quality of the biomass of the host plants. Okay. It will alter the tissue quality and the fitness of the herbivore is the pest. Okay. The trophic level 1 is trait of growth of level. I said, if we put higher end, you have carbon and nitrogen's the ratio need to be balanced in the plant. So when the input higher nitrogens, that means they have a natural defense of this carbon-based defense, like phenolic, tannin, lignin are also affected. Yeah. Okay. And this one, you can see nitrogen is higher, so biomass is higher. So this is when nitrogens, excess of nitrogens with the defense mechanism are bitter. This because this is defense components on the plants needed. So you just see, show here what happens when talking about the optimal nitrogen application and excess nitrogen applications. We have found that, of course, the growth will increase. Yeah. This optimal and excess nitrogens are different. Optimum is when the that's the best levels of the nitrogen needed. But your excess basically doesn't mean it's going to be increase the yield. So, and what see the physical difference in here, what we need here is a physical defense of the plants basically is reduced where we have an excess of N. Okay, and this is also the chemical, the phenolics and tannins acids also will be reduced, suppressed. Okay, then we have to have herbivore fees. We have found that there is, of course, it's fastest the growth of the plants. It's also faster increase the cycle of increase the generations of the pests. And also, and then we go for the third level. This is the impact of the nitrogen VOC. So I will just now this is to show how it alter the relations of phytophic interactions of the plants, predator, and is plants herbivore and parasitoid. So here I will just want to share with you the studies we have conducted. Yeah, we did studies on the nitrogen fertilizations of the cabbage and its natural enemy. So we did the first level, we grows the cruciferous plants, yeah, with the different nitrogen level fertilizations, and then after that we did DBM bioassay, yeah, on the so we track the fecundity or the DBMs, yeah, on the different effect with the different cruciferous fertilized with different rates of the fertilizer and then the third part is we do the of course from the week 1 and week 12, the higher end give the high levels of the biomass. The leaf increase, no doubt, that's common. And then, but the actually by the week 6 actually is that the optimum of the growth of the leaf. And then the dilutions, we did treated the GSL content, actually it already started remobilized after week 6 or so. And then by week 12 we found the defense collapse, yeah, because GLS also reduced when the treatments is increased. Okay, So this is talking about just now the plant. Now we look into the impact on the lava. Sorry, I don't know why it keep moving, but it's like because it's go as a I have to go back here. Effect of the DBM lava longevity. We found that well, the DBM that fed with the higher concentrations nitrogen have a longer life, yeah, and then it's significantly longer life, and this is explained because it's proven that it's increased the nitrogen, basically the affect the plant tissue and plant physiology, and at the same time, therefore, it's also increase the fertility of the increase the longevity of the DBM. And then we have increased compounded adult. We can just, I choose this is just to summarize. We have that with the life cycles, not only is live longer, it's also reduce the life cycles of the larva of the pest. Okay, but this is what we can see that when the larva fed higher nitrogen, the what I call the larva stage is shortened 4.38 days. Yeah, while the its lifespan is 7 days higher. So compared to this one is and the diet. This is longevity of the larva. And we did the parasitism, but this parasitism test we did choice and no choice. We put a difference. First experiment, we give all the with the different plants with the different fertilized with different rates. Another one is just as the same rate. So we found that there is because the percentage greatest with the higher nitrogen treatments. So that means pests, the Poticia will attack higher on the DBM that fed with the higher nitrogens. Okay, and then this is also we say about the developments of the parasitoid itself. So moving again, I'm sorry. Okay, the lifespan of we found that not only the effect of the high nitrogen increase the lifespan of the DBM, but also it increased the lifespan of the Poticia. That means the Poticia that fed with the DBM fed with higher ends. Okay, so but no choice but if talking about no, I would say no choice and choice. They didn't have much difference. There's not much significant difference, and because they feed anything that with nitrogen. They have either it's lower end or higher end. So, but our experiments say there is no significant difference. Yeah, but because this is no choice, and because we put all the same rate. But this one we put at the different rates of the and fertilizers. DBM fed with the different fertilizers. So talking about cocoon development of the Cortesia that fed on the Flutella DBM fed with the different nitrogens. We found that the higher nitrogens, those fed with the high nitrogens have a higher number of the cocoons productions. Yeah, this is found is significant compared to its no treatments and lower treatments. Okay. But actually, I forgot to say that when we found the study here, the optimum rate of the nitrogen is 150 milligram. Okay. So the success rates, of course, the success rates of the cocoon when with the highest end. So this is just some summaries about the parasitism rate is for the choice that is 50%, by no choice at 42%, and then also both big success at 30 milligram, yeah, of the highest nitrogen, and then fertilized, we have higher fertilized successful fertilized potassium, yeah, become cocoon, and the high nitrogens treatments. So this is a summary. The impact on nitrogen fertilization. Can see impact on plants is increase the cabbage leaf area, herbivore, yeah, increase the growth rate, and even shorten larva stage, means the cycle increase, yeah, and then not only increase the cycle, reduce the cycle, but increase the longevity of the insect. And this fertilizer, this nitrogen actually increase pest and parasitoid life, and both parasitoid and the herbivore life. So, and this is just to summarize. Is very important when we look into the management of the pests in the farm. Is not only thinking about how to kill the pest, but we have to start from the beginning for of the whole program of the crop management, because and it started from how we feed our plants. And when you're talking about nitrogens, with putting nitrogens, it's not only because it's a cascading impact. Yeah, it have the future impact of the higher nitrogens fertilizations. And okay, so with this also we have to learn about how to use these practices to manipulate the ecology. So we need to know how to balance it with the agriculture system for our crop protection management in the farm. So I think that is all from me. Thank you very much for listening for my topic. That's all. We cannot hear you, Dr. Henry.
Pak Henry, still mute.
Okay.
Okay.
Okay. Thank you very much, Professor Norida, for your nice presentation. Yeah. Next, we continue to Professor Agustin to present his paper. Please, time for you.
Thank you. Please, my slide is here from the screen. Okay. Still mute, Pak Henry.
Okay, please.
Okay.
You can continue.
Thank you for inviting me as the one of the keynote speaker for discussing today. So as the Pak Henry said, I will discuss about the mealybug. It's still mealybug. Due to the insect is very disgusting over the agricultural production. As we know, mealybug is the one of the scale insect. So actually there are so many scale insect, and the one of the family is Pseudococcidae that we know as mealybugs. And the insects not only attack directly to kill the plants, but also transmit some virus or disease, and making the agricultural condition is making worse and worse. In Indonesia, the study of the mealybug is still limited, and it's also still scientific working on the mealybugs. And this presentation and not only give the knowledge about the mealybugs, but we also calling the students and also researcher for together researching the topic in the mealybugs. And we know the Indonesia is very unique country, and we have so many islands here, the small, the bigger islands, and based on the ecological characters, Indonesia divided into three parts. It's come the Asian fauna, near to the Asia actually, Sumatra, Borneo, Java, and Bali. But other islands such as Sulawesi and Nusa Tenggara, Lombok is the different ecosystem. And we have also the last one after the Walase, we have the Weber Lines, and it is also totally unique. In this situation, the ecosystem will be very huge, and biodiversity is also the largest after the Brazils. It means maybe some species will be benefit, but others could be danger for these islands and also for over the world. So I will try to give the small opinion about the mealybug biodiversity in Indonesia. Hopefully, it will be invites all the students and the scientists and lecturers and others to be focused on aware and learning or studying in the mealybugs fields. As we know, mealybugs making problem not only for shocking pest insects, but also because the colonize the fruits, the plants, making not really attractive. So we can see when we buy something in the markets, and we always find to the beauty fruits and other horticultural products, even though maybe the quality is the taste is the something about related to the quality is good, but the appearance is still being one of our opportunity to choose. In this case, mealybugs will be problems, and direct attack of the mealybugs is it will be impact to the plant directly and reduce the productions, and also can be kill the plants in certain times. The mealybugs can rear every 30 days and produce 1,000 of the eggs, and some of the mealybugs also not laying the eggs, but they release the new nympha as the first stage that can be spread rapidly. We calling the crawling stage, and it is the fatal stage when the farmer can handle the problems. So the mealybug will be spreading fastly, and the size is also very small. It means the wine will also can spread the mealybug after the storms. And now we see there are so many like terminology about the invasive species, maybe alien species. It is related to the mealybugs that the species can be spread sporadically, amazing, without any prediction. And here, let me see, the mealybugs also transmit the virus in pineapple, for instance. The producer will be like difficult to handle these problems due to the position of the mealybug is very difficult to gain. For instance, they can life into the button of the roots, and between the leaf, they also can be separates, and this condition will be getting worse and worse after some symbionts, like the ants, will transport the mealybugs from the others plant to other plant, from the field to other field. We can see the ant is the predator actually, but in these symbionts, the ants will be like helpers for the mealybug for going to spread to others islands. We know the ants could be touch our body, our legs, our anything before we go to the airports, and they will be carrying the mealybug with the ants. The second one, the ants will be like protector for the natural enemies. So when we see there are so many ants around surrounding the mealybugs, it means they didn't kill the mealybug, but they be the antagonistic. They are like bodyguards. They're carrying the mealybugs and kill the natural enemies. And the last one, it is more important things. The ants get the honeydews, and they also clean it. So a honeydew is like the waste. Mealybug can trap into the honeydew. They can move because the sticky liquid from the honeydew. But in this case, the ants will be cleaned. They consume the honeydew and make the waste will be out of the mealybugs. When the populations come with the ants, the population of mealybug will be double or maybe sigmoid. But without the ants, the population will be getting lower. In this case, we have to know and also educate the farmers how the ants influence the mealybug population. In other ways, mealybug is not only getting problem for our life, some of the mealybugs also benefit. For instance, we see the mealybug attack in the cactus that we know as the coccineal insects is produced, and now we see this is the expensive products. It's come from the mealybugs, Dactylopus cactus, because the color is very attracting, very red and amazing. They will stay in the long life position. So when we touch the mealybug in this color, our hand can be getting red in the few days. It means it is organic colors, and now they use for many purpose. The other ones, the mealybugs also majority as decomposer. So when we see mealybug attack the plant, it's only about 40%, but 60% others is decomposer. It means the mealybug is very help us for decomposing the material, especially for organic materials. And now let me see, there are some family from the scale insects that actually they are not the mealybugs. That we see in here, you can see the rows. The mealybugs is so by the rows, and other is looks like mealybugs. So in here, there's the SPDD, and we try to find it in the Indonesian ecosystem or biodiversity. We see the first one is the SPDD, is the highest number of the species, not only in Indonesia actually, but the pattern is also can be applied for the world. So over the world, the SPDD is number one species, the highest species in the scale insects. The second one is Pseudococcidae. And now the number of species getting developed and come to 121 species, belong to 33 genera. the species and also the host of the species can be accessed in the website. And later on, I will show you the website that we built for searching for making the map distribution about the species of the each species of the mealybugs. And here, around 4-year study of mealybugs, we collect 7 new species of the science and 12 new record for Indonesia. Almost the species attack in the horticulture, especially for the fruits. It is true the herbaceous plant is the rare one, but the most one attack on the fruits. We start the research in 2019, and now going on, and we develop a museum as well. So all of the collection we put in the museums, and one by one, maybe in the easy way, I will describe the species for the audience. This species attack the parasitic plants. It means the plant is going to attack the other plants. So in this case, this species could be used for controlling the parasitic plants. We try to find the species for the plant of the parasitics, for instance, avocado and mango and citrus, but unfortunately the species only attack the parasitic one. It means the species could be like
the species only attack the parasitic one. It means the species.
And it means for the next study, it will be like a candidate for controlling the parasitic plant in the productive or agricultural plants. The second one, it is the Polypagus species mealybug that attacks so many fruits from the durians, the harder skin, to the herbaceous skin. For instance, like the water apples, star fruits, and others. And now the last one, we also found that this Mycoccus zenepe attack on the salak. Salak consists so harder skins, but they still can attack this fruit. It means this Mycoccus zenepe will be more danger for the next. The distribution of zenepe with tanks is only around in the Sumatra and Kalimantan. It means we have to save other island for infestating the species to other islands. So it is important for the quarantine maybe to keep the species not distribute around other island or maybe other countries. The next one, we also found the Planococcus bengkuluensis is only attack on coffee. It's still monophagous species. It's a good one because they can't live another like plants, and it's special in the coffee, but it's still maybe have the problem for the next if the species maybe invite the others country that also plant the coffee. So it is new information maybe for the coffee lovers. The next one is Pseudococcus ifahe, attack in the peperaceae. There are so many peperaceae products in our country and also the other country, and we found it in the like pepper beetle, pepper adangtum, like it's also we call it lada maybe in here. It is the most good product for the peperaceae plants. And the next one, this one, we found it in the pineapple, Planococcus sonoi, is attacked on the pineapple. again, it is like yeah, the deforestation for the next uh, pineapple plantation maybe, because they're spreading a lot of in Bengkulu. But another one, maybe we can say that because it is an endemic species, maybe there are so many natural enemies come in this area. Later on, I will see you how to control the mealybugs, the endemic mealybugs, especially in natural enemies. The last one, Paraputora fei and Paraputora martonei, but we found it not in the horticultural product, but in the forestry. It means it could be attack some fruit like mangosteen, like durian as well, but we still find it in limited area. And other one is a new record species. It is also attack on the parasitic plants. It is in the forestry. It's also in the forestry, especially on furniture plants, woody plants, and it is for also the butternuts. We find it is as the new record, not the new species, but already record. Interestingly, the species also we found in India. Some maybe like hypothesis maybe why we found it in Bengkulu and we also find it in India, for instance. In this case, we know Indonesia, especially in Bengkulu, we was colonized by the British at the times, and the transportation between British and India is very excessively at the time. It means it could be the species come from India is come from Bengkulu, or revised. The species that we know from Bengkulu is come from the India. It means the transportation making the distribution clearly. And here some picture drawings, pictures, scientific drawing picture that we made, and it is clear that some the species is new record and new species that our library. Now this is the big data that we build. We can access by online indomealybug.com. All of the species we put here, the information, host, and also the distribution. So maybe the student, the others can use this website for reference. And now talking the controlling the mealybugs before Dr. Norida also using this technology in the lab, and others also use it. We put in the 3 section: preventive, monitoring, and also the curative, maybe. And I will not describe all of the items because it's very general, but I will try. I want to describe more specific, deeply about the strategy in biological control. For controlling the mealybugs, the good way that we found until now is using the predator and parasitoid. Otherwise, entomopathogenics such as Beauveria bassiana maybe, and some nematodes is also can control. But for classical biological control, mostly Cryptolemus mantaroides, the good way for control. Another one, we can use the volatile oils maybe, like essential oils come from the fruits and other oils, because mealybug consists of the so many fat and mealy around the body, so they can they can be erased by the essential oil. The other one now is going bigger and bigger, the technology. They're using the herbivore-induced plant volatile, for instance, and also using the biopeticide, using the activity from RNA interface based treatment maybe, and now is still evaluated by the researchers. I think it's all for discussion our time for the second time, and hopefully if you have many information or maybe want to know some information from us, I think it's a good way. Thank you very much, Professor Norida, and maybe other can give also background from your sides and also the participant. Thank you, Pak Henry.
Okay, thank you for Professor Agustin Darkani. I think a nice presentation with new inventory, yeah, and new record. Okay, next we go to Professor Amelia, yeah, from Philippines. Hello, Amelia.
Hello, Dr. Henry. Good afternoon. Before my presentation, I'd like to thank the organizing committee, most especially you, Dr. Henry, for giving me this opportunity to be part of the second ISPP. Actually, I have prepared a pre-recorded video presentation. In as much as I would like to have a real-time presentation today, but my current situation wouldn't allow me. If I will still push through with real-time presentation, I'm afraid that it might get cut. My presentation might get cut at the middle or where else, because today we have a power interruption in my area. Luckily, I have found us an area where there is electricity, but internet connection is quite weak. From time to time, there is this message that pops up in my computer, my laptop, saying that my internet is unstable. So with that, please bear with me. Last night, I prepared this video presentation for you, but I will stand by for the Q&A during the open forum. May I request the organizing committee to please present or play the video presentation. Thank you very much, and thank you. Good morning, everyone. Let me begin with a simple question. When we hear the words plant protection, what immediately comes to our minds? Is it pests, diseases, fungicides, insecticides, or resistant varieties? These are, of course, important, but today I would like to ask a different question. Where are the pollinators in our plant protection programs? Where are the bees? Where are the natural enemies? Where are designing resilient agro-ecosystems? Let us begin with our Good morning, everyone. Let me begin with a simple question. When we hear the words plant protection, what immediately comes to our minds? Insect pests, diseases, fungicides, insecticides, or resistant varieties? These are, of course, important, but today I would like to ask a different question. Where are the pollinators in our plant protection programs? Where are the bees? Where are the natural enemies? Where is biodiversity? Where are the ecological processes that quietly make agriculture production possible? Because perhaps the future of plant protection is not simply about becoming better at killing what harms our crops. Perhaps it is about becoming better at designing agricultural systems that are naturally more resilient. And this is where I would like to introduce the concept of pollinator-centered horticulture within a One Health framework. We cannot have healthy horticultural systems without healthy agro-ecosystems. And we cannot have healthy agroecosystems without healthy pollinators. My presentation today is entitled Advancing One Health Through Pollinator-Centered Horticulture. This topic brings together three areas that are often discussed separately: plant protection, pollinator health, and One Health. My argument is that this should no longer be treated as separate conversations. If we want sustainable horticulture, we need to consider the health of the crop, the health of the pollinator, the health of the environment, and ultimately the health and well being of people. So today, I would like to take you on a journey from controlling pests to protecting ecological services to designing resilient agroecosystems. Let us begin with our traditional understanding of plant protection. We protect crops against insect pests and plant diseases. We use fungicides and insecticides. We develop and deploy resistant varieties. These approaches have contributed enormously to agriculture productivity. So I want to be very clear. I am not proposing that we abandon plant protection. The question is much more fundamental. How do we practice plant protection without compromising the biological systems that agriculture itself depends upon? Because a crop is never alone in a field. A tomato plant, for example, is part of a much larger biological community. There are pollinators, there are predators and parasitoids, there are microorganisms, there are soil microorganisms, there are surrounding plants, there is water, soil, climate, and most importantly, the farmer. Therefore, when we protect a crop, what else are we protecting or potentially harming with our intervention? And this question leads us to our first major shift. Traditionally, our attention has been directed toward the problem. Where is the pest? What disease is present? What pesticide should we apply? But the slide proposes a different way of thinking. Instead of focusing exclusively on the problem, we begin to focus on the health of the system. We ask, are pollinators present? Are natural enemies present? Is biodiversity maintained? Are soils healthy? Is the landscape resilient? This is not a rejection of pest management. It is an expansion of pest management into agroecosystem management. And this distinction is important because a pest does not exist independently of the ecosystem. If we remove one organism, we may also affect organisms that regulate it. If we simplify the landscape, we may remove resources needed by beneficial insects. If we expose pollinators unnecessarily to pesticides, we may compromise an ecosystem service. Therefore, plant protection in the 21st century must evolve from controlling pests to managing healthy agroecosystems. Now, let us imagine that agriculture can move in two different directions. On the left, we see a cycle of decline and dependency. Pest outbreaks lead to more pesticides. Greater pesticide pressure can contribute to pollinator decline and loss of beneficial organisms. Reduced ecological balance can then contribute to greater vulnerability, and the system becomes increasingly dependent on external inputs. But on the right, we see another possibility. We begin with biodiversity. Biodiversity supports healthy bees support healthy pollination. Healthy pollination supports healthy crops. Healthy crops contribute to resilient farms, and balanced ecosystems can contribute to reduce pest pressure. Ultimately, we connect this with healthy people, a healthy environment, and sustainable livelihoods. So perhaps the question we should be asking is not how much pesticide can we use safely, but also how can we design the agricultural system so that we need fewer corrective interventions in the first place. This brings us to the paradigm shift. The old paradigm says protect crops from pests. The new paradigm says protect crops and protect the ecological services that sustain crop productivity. The second part is critical because agricultural productivity is not produced only by seeds, fertilizers, irrigation, pesticides, and machines. Nature is also working for us. Pollination, biological control, nutrient cycling, soil processes, water regulation. These are ecological services. And if our plant protection strategy destroys these services, then we may solve one problem while creating another. Therefore, the mindset must shift from doing less harm to creating more ecological health. This is the foundation of the argument I am presenting today. And this brings us naturally to One Health. One Health tells us something very fundamental. Health is interconnected. We cannot completely separate human health from plant health. We cannot completely separate plant health from animal health. And we cannot separate any of this from environmental health. Let us look at the four components. One, people. We need food security, nutrition, health, and well being. Two, plants. We need healthy crops, productivity, biodiversity, and sustainable horticulture. Three, animals. And here I want to emphasize our pollinators. Their health, diversity, and ecological functions are part of agriculture health. And finally, the environment. Healthy soils, clean water, biodiversity, climate stability. These components continuously interact. Therefore, when we talk about plant protection, we are actually talking about a much larger health system. Plant protection becomes part of One Health. So, I would like you to consider this question. If a plant protection intervention protects the crop, but damages its pollinators and the surrounding ecosystem, can we really call that a healthy agricultural system? That is the question at the heart of One Health agriculture. Now, we arrive at the heart of my presentation pollinators, particularly bees. Pollinators are often discussed in the context of biodiversity conservation, but they should also be discussed in the context of plant protection and agricultural productivity. Pollinators contribute to fruit set, seed production, fruit quality, uniformity, shelf life, and nutritional quality. And look at the diversity represented here. Honeybees, bumblebees, stingless bees, butterflies, and other pollinators. As someone working with stingless bees, I have become increasingly convinced that we should not look at these insects simply as organisms that visit flowers. We should look at them as partners in agriculture production. And when we protect these partners, we are not only conserving biodiversity, we are protecting an ecosystem service. But pollinators are only part of the story. We also need to understand pollination itself. Pollination is much more than transferring pollen. It determines whether reproductive structures successfully develop. And as shown here, it influences fruit set, seed production, fruit quality, shape, uniformity, shelf life, and nutritional quality. So inadequate pollination, that does not necessarily mean simply no fruit. It can also mean poorly developed, deformed, less uniform, or lower quality produce. From a horticultural perspective, this matters tremendously because farmers are not only selling kilograms, they are selling quality, they are selling appearance, they are selling uniformity, they are selling marketability. Therefore, pollination should be treated as an integral component of horticulture production, not as an incidental ecological process. Now, I want to make a stronger proposition. Healthy pollinators do not stop at pollination. They are part of a pathway toward crop resilience. Look at this sequence. First, pollinator health. Healthy pollinators need adequate food, clean water, nesting resources, appropriate disease management, and reduced pesticide exposure. Second, effective pollination. More flowers successfully pollinated, better fruit set, more seeds, more consistent pollination. Third, healthy plants, better development, stronger growth, improved allocation of resources. Fourth, crop resilience, greater productivity and stability. And finally, resilient agroecosystems. But there is another important feature of this diagram. At the bottom, we see the threats to pollinator health: pesticide misuse, habitat loss and fragmentation, climate change, monoculture and poor floral landscapes, diseases and parasites. And these are not problems that a beekeeper can solve alone. These are landscape problems. Therefore, protecting pollinators requires collaboration between plant protection specialists, farmers, ecologists, beekeepers, communities, and policymakers. The health of the pollinator is ultimately connected to the health of the landscape. So, what does this mean for plant protection? On the left is conventional plant protection. The primary focus is eliminating pests and diseases. The approach may involve synthetic pesticides, broad spectrum control, and calendar based applications. On the right is pollinator centered plant protection. Notice that we have not removed pest management, we have changed the context and the objective. We use IPM. We use selective and judicious pesticide applications. We conserve habitats. We support pollinators and natural enemies. We monitor populations and make threshold-based decisions. And what do we gain? Healthy pollinators, better pollination, better crop quality, greater biodiversity, natural pest suppression, more resilient agroecosystems. So let me make this distinction very clear. Pollination centered plant protection does not mean never use pesticides. It means use pesticides intelligently, selectively, and only when justified, while protecting the organisms and ecosystem services that agriculture depends on. Now, let us make this practical. How do we actually implement pollinator-centered plant protection? We begin with monitoring. Don't spray because it is Monday. Don't spray because it is a flowering season. Scout first. Identify correctly. Determine whether intervention is actually necessary. Second, use cultural practices like crop rotation, diversified cropping, intercropping, healthy soil management. Third, conserve pollinators and beneficial organisms, provide flowering resources, maintain hedgerows, protect habitat. Fourth, use pesticides judiciously. Fifth, consider timing. If pollinators are actively foraging, this matters. Sixth, protect nesting habitats. Seventh, strengthen biological control. And finally, evaluate and adapt. This is the essence of IPM, decision making based on evidence, monitoring thresholds, and ecological understanding. And I particularly like the principle at the bottom: work with nature, not against it. But if we are serious about this transformation, we need to ask, what should we be researching next? I see several exciting frontiers. First, AI and smart technologies. Can AI help us monitor pollinator activity? Can sensors detect changes in colonies? Can digital tools support pest and disease decisions? Second, microbiomes. What microorganisms are associated with bees? What microorganisms occur on flowers? How do these microbial communities influence pollinator health and plant health? Third, genomics and omics. Can we understand genetic adaptation in bees? Can genomics help us develop more resilient crops? Can pathogen genomics help us detect threats earlier? Fourth, climate adaptation. How will changing temperatures, rainfall patterns, extreme events, and landscape changes affect pollination? And how do we design landscapes that remain functional under climate stress? These questions require something beyond disciplinary research. They require collaboration. Entomologists cannot do this alone. Plant pathologists cannot do this alone. Beekeepers cannot do this alone. We need science, innovation, and collaboration. So, where do we go from here? I would like to propose a small change in the questions we ask. Instead of asking only how do we eliminate pests, let us also ask how do we conserve pollinators? How do we restore biodiversity? How do we strengthen ecosystem services? How do we design resilient agroecosystems? These questions do not replace our traditional plant protection questions, they expand them. And this is the transformation I am proposing from controlling pests to designing resilient agroecosystems. The first approach asked, how do I remove the problem? The second asked, how do I design a system that can remain productive despite problems? That means conserving biodiversity, protecting pollinators, building resilient farms, designing resilient landscapes, and creating sustainable food systems. This is a fundamentally different way of thinking about plant protection. We move from reaction to prevention, from simplification to biodiversity, from dependency to resilience, from controlling individual organisms to managing ecological relationships. Let me bring everything together. At the top, we have One Health, people, plants, animals, environment. This lead us toward sustainable horticultural systems. Within those systems, we need to protect three interconnected dimensions: pollinator health, plant health, environmental health. Ladies and gentlemen, I would like to leave you with one thought. The future of horticulture will not be determined only by how effectively we control pests and diseases. It is also be determined by how successfully we protect the ecological processes that sustain crop production. Pollinators are one of those processes. Bees are not simply visitors to our flowers, they are partners in our food systems. Therefore, protecting pollinators is not separate from plant protection. It is part of plant protection. Protecting biodiversity is separate from productivity. It is an investment in resilience. And One Health is not simply a framework for thinking about disease. It is a framework for designing the agricultural systems of the future. That ends my presentation. Thank you very much for your attention. Good morning, everyone. Let me begin with a simple question. When we hear the words plant protection, what immediately comes to our minds? Insect pests, diseases, fungi.
Okay, thank you. Yeah, thank you very much for Professor Amelia. I think the very interesting presentation. Yeah. Now we go to discussion. Yeah. First, we go to Professor Norida. Yeah. You can answer. Yeah, answer the question from Miftah. Yeah. Miftah from Research and the Research Center for Estate Crop Print. Yeah. the question, his or her.
Okay,
Question.
well, thank you. Well, I read the question from Mita. Thanks for ask the questions. Okay, I think this result of the especially parasitism rate, yeah. So far, when we see our study is quite high, the parasitism rate for the high nitrogen fertilizations. I think this can be make it more. We can do this study further because we haven't find any optimum rate yet for the parasitism. And just to be, if you want to apply, of course, you say this going to be have a problem. If you say, oh, we want to increase the parasitism rate in the field, so apply the nitrogen. But we have to be aware if we apply nitrogen, high nitrogen, it also increase the I call it the DBM production rate. So therefore, I mean, we have to be careful on that, and we also have to, in the field, we have to think about how to increase the fitness and of the parasitoid in the field. Yeah? So it can be it can attack it can produce higher, where it can attack the DBM, control the DBM actually. So I think this is also, I think we have can do more further study, right? We increase more substitute plants where we can for fitness of the parasitoid itself. Yeah, and one thing is this study, basically when we did this, actually because we want to increase one of the objective of this study, we want to increase the productions of the Cortisia itself. So if you are doing in the lab, I think it will be good result. So it can have a higher Cortisia populations. You know. Okay, is that answer the question, Mifta? Sorry.
Okay, thank you for Professor Norida. Now we go to Professor Amelia. Yeah, woman first. Yeah. This one question from Miftah. Yeah. About could you provide an example of a study related to maybe this pollinator? Okay, please answer.
Okay. So we have an ongoing study on the genetic structure, population genetic structure, and genetic diversity of Tetragonula beroi. Tetragonula beroi is the widely cultivated stingless bee species in the Philippines, in my country, and it has a high economic potential. That's why we are really into it. We are really developing the stingless bee keeping industry in the Philippines. And related to your question, that is one example of the study related to omics and genetics that we are doing. So by collecting different samples from all over the country, we would know whether these bees from different places are genetically different. And we can also determine whether the populations represent genetically distinct lineages and could provide evidence for conservation units. So that is one study that we do in relation to this. So I hope that answers your question. Ma'am Mifta.
Mr.
I missed it. Sorry for that. Thank you.
Thank you, Prof. Amelia. Next, we go to Professor Agustin. You can answer 3 questions.
Okay, thank you, Pak Henry. The first one is come from
Sylvie.
Bu Miftah. Yeah, Miftah. The first one is Miftahur Rohmah. Nice one. So Mostly, mealybug is polyphagous species that attack so many plants, host plant. But in our library, we still also find specific or polypha, not the polypha, but the monopha. It means the limitation of the host plant is could be like we less of information, not because the monopha only, but we lack of the information maybe. But for the Oryzococcus ozeri that we found attack on the parasitic plants, we walking around 4 or 5 years, we still didn't find the species except on the parasitic plants. We try to investigate all of the part of the plants that the parasitic plant is host on the plants, but we didn't find it. We cannot say it is totally specific or monopha before we found the species to other host plant. So in this case, we still stay in the statements. It could be specific or monopha, but it's still running. The information is still running, and we also now working on the other side of Indonesia, in the Walase, in the Webers. In this year, we working on the Maluku. It is located in the Morotais, around the Morotais, is near the Philippines. Now, maybe later on we can see how many species in the Morotai is also related to the Philippines. So it's still going. The second one is come from Bu Sylvie. Okay. Mostly we identify the species using the morphology character. So not using the moleculars. People ask how you just working on the morphology. We also working on the molecular actually, but due to the methods and also we say the easy one is morphology. But when we face the problem between the close relative in the morphology, because we know there are so many polymorphism species, now we solve by molecular. So up to that, we are working on the morphology as the good way for identifying the species. And the last one from the Bu Norida. Bu Norida, the species that we mentioned before, it is Pseudococcus Sosormarsonei. It is attack on the pineapples, and also up to date we just found it in the pineapple. So there are so many samples we got now, around more than 5,000 specimens we have in our labs, but we still found that it is only attack on the pineapples. We don't know. It is maybe associated a long time ago because it's endemic. It is not alien species. It is also not invasive species because we just found in this island, in the Sumatra Islands, especially in Bengkulu and Enggano Island. Enggano Island is the lowest islands in front of the Australia, but we still find it in this host plant. It's no problem actually because the population is low. The question comes for the next, why is the population low? Because in the mix there are so many natural enemies they found here. We last semester we found two specific parasitoid. It's come from the Anagyrus species, and we still debate why the population is low due to the endemic, or maybe the species is not quite developed well because the limitation of the host plant. And now the easy way to control the mealybugs in the pineapple, they try to put the plants before planting in the area using some chemical treatments, or maybe temperature mechanical treatments like temperature, or maybe they try to make sure the plant variety of the plant is stronger. It means resistant from the mealybugs. Now 2 years ago that I not missed it, the pineapple from Indonesia is banned to export to overseas due to the mealybugs in around the fruits. Mostly Indonesia export using the can. It's not the fresh one, but canning fruits, or maybe now it's like dry fruits as well. So the distribution of the mealybug is still can be limited by some regulation. I think it's all of the question. Thank you. And I'm also actually waiting for your suggestion for the participants. And next discussion can be whether using the email or maybe media social. We also have the media social like Instagram, and we put the mealybug Indonesia mealybug as the title of the media social. Thank you very much.
Prof. Henry, Dr. Henry, may I say something? I have actually a question for Prof. Agustin. Your presentation about mealybugs. In one of the provinces here in our region, we have a very big problem on mealybugs, and we have a large plantation of pineapples here. The team working on that have already, they said that they've already identified the species of the mealybug, but after a while, they are now in doubt whether that is really the correct identity of the pest, because they've already tried several measures, several strategies just so to control that, but again and again they failed. And I think may I ask or request from you, Prof. Agustin, that if I may get your contact details after this session, so I can connect you to the team of from the Department of Agriculture. They are really having a big problem on mealybugs in pineapple.
Okay, thank you, Prof. Amelia Nicholas. It is benefit of the seminar, so we can tie the networking after the seminars, and of course, yeah, we can contact after that, and we also have the research gates accounts and also the Instagrams, or maybe we can contact by WhatsApp or line, etc. Thank you.
Yes.
We are waiting for your contacts and we can discuss about it.
Okay, thank you, Prof. Agustin. Thank you so much.
Welcome.
Okay, thank you very much for Professor Agustin and Professor Amelia. I think there is some question or discussion. Now from Bu Sampurna Ginting, please. Yeah.
Okay.
Unmute. Okay. Hello, host. Can you unmute Bu Sampurna?
Okay. I will ask for Professor Amelia. I am Sampurna Ginting from Department of Plant Protection, Faculty of Agriculture, University.
Ma'am, The audio is not so clear.
Okay, I am
Yeah.
Sempurna Ginting from Department of Plant Protection, Faculty of Agriculture, University of Bengkulu. A very interesting your presentation. I will ask you about how we can change the mindset of the farmer who are already attached to pesticides to control the pest and disease. How we return to organic farming for sustainable agriculture. Thank you.
Okay, thank you for those two challenging questions. That is really, that still remains a challenge, especially to us from the academy. We do research, we do extension work, and how do we convince them to shift from using chemical pesticides to doing like organic farming or lessening the use of chemicals. What we do now, I will answer your question, ma'am, based on what we are practicing here. So because I am working on stainless steels, when we do extension work, we explain to the farmers the benefits of bees to them, to the environment, to biodiversity, and especially to their health. So if we can connect that to the health and to the benefits that they can gain from like raising or culturing stainless steels, for example, they really think about it. And some of them slowly, little by little, are, though I am not saying that it's really easy, it's really tough to convince them, especially if they are already, they have this fixated mindset that when they use chemicals, it will, easily solve their problems on pests. So what we do, which is, I think, quite effective in a way, is we tie it, when we explain, when we show them, we tie it to the benefits of the health and to the other advantages that they can gain from like taking care of the pollinators like me. So I know that everywhere in the world is having a problem like that. I hope everybody would agree with me on that. It's really challenging. It's really like you cannot really impose them to do IPM, to practice IPM. But if we can like explain to them the disadvantages of that, most especially to their health, what kind of produce will sell, and what kind of produce they will make their family eat, then they get to understand. I think we really just need to be very, very intentional, be very, very patient in explaining to them, letting them understand. Because I believe if a person fully understands something, that person will have a paradigm shift, or we can help them change how they view things such as the use of synthetic chemicals to crops. Just like what I have said in my presentation, that we cannot really, avoid using chemicals, but we can somehow lessen it we can lessen it. We can be very, very intelligent, intentional in using chemicals. But if our target is to completely eliminate the use of synthetic chemicals, that for me is impossible. So I think what we can just tell the communities, the farming communities, the farmers, is to just at least somehow lessen the use and have make it judiciously, practice the use of chemicals. So I hope, ma'am, I answered your questions regarding that.
Okay, Professor, thank you.
Thank you for your time.
Okay, thank you very much for all keynote speaker. Our time is over. Yeah, I want to read the conclusion. Yeah, first, yeah, that's very important. Yeah, important to manage the nitrogen. Yeah, because nitrogen can increase the production, plant production, also increase the parasitism, and longevity. But application of nitrogen also increase the faster larva development and adapt longevity. So we must obey, we must obey, and important to manage the application of nitrogen. And second, mealybug, mealybug also is very important. First, they attack various plants, and act as virus vector, the process of infection, symbiosis by N. But mealybug also have a benefit. One of them, they produce a natural red food for medicine or food coloring. And in Indonesia, the professor found 7 new species and released 12 new records. So there is potential to develop the research in mealybug. And then 3, from Professor Amelia, there is 1 health framework, to move from conventional plant protection to pollinator center plant protection. We can apply the pollinator friendly IPM strategy by move from controlling pests to designing resilient agro ecosystem to sustain the agriculture. I think that's all. To end this session, let we give applause for all keynote speaker. Applause to Professor Norida, to Professor Agustin, and to Professor Amelia Nicholas. Thank you very much for your contribution in this seminar. Thank you.
Thank you, Dr. Insinyur Hendry Bustamam MS, for moderating in second session. We also sincerely appreciate Dr. Norida Mazlan, Professor Agustin Zarkani, and Professor Amelia Nicholas for their insightful and valuable presentation.
Ladies and gentlemen, As a form of our sincere appreciation, we will now proceed to the symbolic certificate presentation for the keynote speaker and moderator.
The first certificate of appreciation is present to Dr. Norida Mazlan from University Putra, Malaysia. The second certificate of appreciation is present to Professor Agustin Zarkani from the University of Bengkulu, Indonesia. The third certificate of appreciation is present to Professor Amelia Nicolas from Central Bicol State University of Agriculture, Philippines. The next certificate of appreciation is present to our Session 2 moderator, Dr. Insinyur Henry Bustama MS, for his valuable contribution in guiding and moderating the discussion session. We express our highest appreciation to all keynote speakers and moderator for their valuable contribution to International Seminar on Plant Protection 2026.
Ladies and gentlemen, before closing the keynote session, we will proceed with the commentation. We kindly invite our keynote speaker, moderator, committee members, and participants to turn on camera if possible.
To the documentation team, please kindly take the screenshot. First slide, ready? 1, 2, 3. For the next slide, 1, 2, 3. We would like to inform all presenters that the parallel oral presentation session will begin at 13:15 Western Indonesian Time. Presenters are requested to enter the assigned breakout rooms according to the final room distribution announced by the committee. Please prepare your presentation file and use the Zoom display name format determined by the committee.
On behalf of the organizing committee, we extend our deepest gratitude to all keynote speaker, moderator, participant, presenter, and invited guests for your valuable contribution to International Seminar on Plant Protection 2026.
We hope today's keynote session enrich our understanding and strengthen collaboration in plant protection, especially in the management of horticultural plant pests and diseases. We apologize for any shortcomings during the program. Thank you for your participation. Wassalamualaikum warahmatullahi wabarakatuh. Enjoy your break and we look forward to seeing you again in the parallel oral presentation session.
Program Studi Proteksi Tanaman atau PSPT merupakan salah satu program studi strata 1 di Fakultas Pertanian Universitas Bengkulu. Visi program studi yaitu menyelenggarakan program studi bereputasi internasional pada tahun 2025 di bidang proteksi tanaman tropis dan ekosistem pesisir dengan fokus pada eksplorasi keanekaragaman hayati serta pengembangan teknologi pengendalian hama dan penyakit tanaman yang berbasis sumber daya alam. PSPT berperan penting dalam pengembangan ilmu perlindungan tanaman. Dalam 5 tahun terakhir, riset dosen PSPT berhasil menemukan 4 spesies baru kutu putih yang berasal dari Indonesia, serta 8 spesies lain tercatat sebagai temuan baru di Indonesia. Data spesies baru tersebut dapat diakses secara lengkap melalui laman indomelibak.com yang kini menjadi rujukan nasional maupun internasional. Temuan lain yang tak kalah penting adalah ditemukannya virus baru pada pepaya Kalina di Bengkulu. Riset karakterisasi begomovirus ini dilanjutkan bekerjasama dengan National Chongqing University di Taiwan. Untuk mewujudkan visi bereputasi internasional, PSPT telah melaksanakan berbagai strategi penguatan tata pamong, tata kelola, dan jejaring kerjasama. Di antaranya, melalui program World Class Professor pada tahun 2022 dan 2023, penyelenggaraan seminar nasional dan internasional, kuliah umum bersama dosen tamu luar negeri, serta keterlibatan aktif dosen dan mahasiswa dalam forum akademik berskala nasional dan internasional. Pada tahun 2023, 6 dosen Universitas Bengkulu terlibat dalam program World Class Professor. Salah satunya adalah Profesor Agustin Zarkani dari PSPT yang menjalin kerjasama dengan Çukurova University di Turki, serta riset bersama University of Malaya di Malaysia. Dosen PSPT lainnya, Dr. Mimi Sutrawati, aktif menjalin penelitian dengan IPB University dan menjadi pembimbing pendamping bagi mahasiswa proteksi tanaman di IPB. PSPT juga mendorong dosennya untuk berkiprah di kancah internasional, seperti Dr. Henry Bustaman yang menjadi dosen tamu di University of Heng Sarmin Teng Bong Mum, Kamboja. Sebagai pengakuan kualitas, PSPT telah terakreditasi International Akwin sejak 2022 hingga 2030. Saat ini, dosen tetap PSPT terdiri dari 14 orang, termasuk 3 profesor, 4 doktor, dan 2 dosen yang sedang menempuh studi doktoral di UGM dan IPB University. Kompetensi dosen didukung rekam jejak penelitian, pengabdian, dan publikasi ilmiah yang sangat baik. Dalam 3 tahun terakhir, dosen PSPT menerbitkan lebih dari 220 artikel ilmiah, hampir setengahnya di jurnal internasional bereputasi. Dukungan sumber daya manusia yang berkualitas juga tercermin pada rata-rata beban kerja pengajaran, penelitian, dan pengabdian kepada masyarakat yang melebihi standar nasional. Penelitian dan pengabdian dosen PSPT tidak hanya menghasilkan publikasi, tetapi juga luaran nyata seperti pendaftaran varietas tanaman baru dan buku ilmiah. Kemahasiswaan PSPT menunjukkan capaian yang membanggakan. Pada tahun 2023, terdapat 112 calon mahasiswa yang mendaftar dan 50 orang diterima setelah seleksi ketat. Hingga semester genap 2023 hingga 2024, jumlah mahasiswa aktif mencapai 194 orang. Masa studi rata-rata mahasiswa antara 4 hingga 4,88 tahun, dengan presentasi kelulusan tepat waktu yang melampaui rata-rata fakultas. Sejak berdiri hingga akhir 2023, PSPT telah meluluskan 89 orang lulusan dengan rata-rata IPK di atas 3,3. Sebagian besar lulusan bekerja pada instansi pemerintah, lembaga riset, perkebunan swasta, dan sektor perbankan. Bahkan, banyak yang menjadi wirausaha mandiri. Beberapa lulusan juga melanjutkan studi magister, baik di dalam negeri maupun di luar negeri, seperti National Chongqing University Taiwan. Dalam mendukung proses pembelajaran, PSPT memiliki fasilitas yang lengkap dan representatif, ruang kuliah modern, ruang kerja dosen, laboratorium riset dan praktikum, hingga Museum Serangga Kutu Putih yang menjadi satu-satunya di Asia Tenggara. Museum ini telah menjadi pusat rujukan peneliti dalam dan luar negeri untuk sabbatical leave, magang, dan analisis spesies. Laboratorium PSPT terdiri dari lab taksonomi serangga, lab nematologi, lab fitopatologi, lab pengendalian hayati, serta lab biologi molekuler yang mendukung riset hingga level genetik. PSPT juga aktif menjalankan program Merdeka Belajar Kampus Merdeka atau MBKM. Mahasiswa berkesempatan mengikuti pertukaran dengan 12 perguruan tinggi lain di Indonesia yang tergabung dalam APSITA, mengikuti kampus mengajar dan berbagai kegiatan pengayaan pengalaman belajar. Penelitian PSPT diarahkan pada pemanfaatan keanekaragaman hayati sumber daya lokal dalam pengendalian hama dan penyakit tanaman terpadu. Selama 3 tahun terakhir telah dilaksanakan 74 penelitian dosen yang diseminasi dalam puluhan seminar nasional dan internasional. Sementara dalam bidang pengabdian kepada masyarakat, PSPT memiliki 2 desa binaan dan puluhan program yang terintegrasi dengan kurikulum pembelajaran. Capaian lulusan PSPT terus meningkat. Sebagian besar alumni berhasil mendapatkan pekerjaan kurang dari 6 bulan setelah lulus, dengan tingkat kesesuaian bidang kerja yang semakin tinggi setiap bulannya. Berbagai keunggulan PSPT antara lain kualifikasi akademik dosen, kurikulum berbasis KKNI yang relevan dengan MBKM, publikasi ilmiah yang produktif, hingga fasilitas laboratorium dan museum yang mendukung pengembangan kompetensi mahasiswa. Sebagai program studi yang terus berkembang, PSPT Fakultas Pertanian Universitas Bengkulu berkomitmen menjadi pusat pengembangan ilmu proteksi tanaman tropis yang bereputasi internasional, menghasilkan lulusan yang kapabel, adaptif, dan siap berkontribusi dalam pengelolaan pertanian berkelanjutan berbasis sumber daya hayati.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Program Studi Proteksi Tanaman atau PSPT merupakan salah satu program studi strata 1 di Fakultas Pertanian Universitas Bengkulu. Visi program studi yaitu menyelenggarakan program studi bereputasi internasional pada tahun 2025 di bidang proteksi tanaman tropis dan ekosistem pesisir dengan fokus pada eksplorasi keanekaragaman hayati serta pengembangan teknologi pengendalian hama dan penyakit tanaman yang berbasis sumber daya alam. PSPT berperan penting dalam pengembangan ilmu perlindungan tanaman. Dalam 5 tahun terakhir, riset dosen PSPT berhasil menemukan 4 spesies baru kutu putih yang berasal dari Indonesia, serta 8 spesies lain tercatat sebagai temuan baru di Indonesia. Data spesies baru tersebut dapat diakses secara lengkap melalui laman indomelibak.com yang kini menjadi rujukan nasional maupun internasional. Temuan lain yang tak kalah penting adalah ditemukannya virus baru pada pepaya Kalina di Bengkulu. Riset karakterisasi begomovirus ini dilanjutkan bekerjasama dengan National Chongqing University di Taiwan. Untuk mewujudkan visi bereputasi internasional, PSPT telah melaksanakan berbagai strategi penguatan tata pamong, tata kelola, dan jejaring kerjasama. Di antaranya, melalui program World Class Professor pada tahun 2022 dan 2023, penyelenggaraan seminar nasional dan internasional, kuliah umum bersama. Program Studi Proteksi Tanaman atau PSPT merupakan salah satu program studi strata 1 di Fakultas Pertanian Universitas Bengkulu. Visi program studi yaitu menyelenggarakan program studi bereputasi internasional pada tahun 2025 di bidang proteksi tanaman tropis dan ekosistem pesisir dengan fokus pada eksplorasi keanekaragaman hayati serta pengembangan teknologi pengendalian hama dan penyakit tanaman yang berbasis sumber daya alam. PSPT berperan penting dalam pengembangan ilmu perlindungan tanaman. Dalam 5 tahun terakhir, riset dosen PSPT berhasil menemukan 4 spesies baru kutu putih yang berasal dari Indonesia, serta 8 spesies lain tercatat sebagai temuan baru di Indonesia. Data spesies baru tersebut dapat diakses secara lengkap melalui laman indomelibak.com dan kini menjadi rujukan nasional maupun internasional. Temuan lain yang tak kalah penting adalah ditemukannya virus baru pada pepaya Kalina di Bengkulu. Riset karakterisasi begomovirus ini dilanjutkan bekerjasama dengan National Chongqing University di Taiwan. Untuk mewujudkan visi bereputasi internasional, PSPT telah melaksanakan berbagai strategi penguatan tata pamong, tata kelola, dan jejaring kerjasama. Di antaranya, melalui program World Class Professor pada tahun 2022 dan 2023, penyelenggaraan seminar nasional dan internasional, kuliah umum bersama dosen tamu luar negeri, serta keterlibatan aktif dosen dan mahasiswa dalam forum akademik berskala nasional dan internasional. Pada tahun 2023, 6 dosen Universitas Bengkulu terlibat dalam program World Class Professor. Salah satunya adalah Profesor Agustin Zarkani dari PSPT yang menjalin kerjasama dengan Çukurova University di Turki, serta riset bersama University of Malaya di Malaysia. Dosen PSPT lainnya, Dr. Mimi Sutrawati, aktif menjalin penelitian dengan IPB University dan menjadi pembimbing pendamping bagi mahasiswa proteksi tanaman di IPB. PSPT juga mendorong dosennya untuk berkiprah di kancah internasional, seperti Dr. Henry Bustaman yang menjadi dosen tamu di University of Heng Sarmin Teng Bong Mum, Kamboja. Sebagai pengakuan kualitas, PSPT telah terakreditasi International Akwin sejak 2022 hingga 2030. Saat ini, dosen tetap PSPT terdiri dari 14 orang, termasuk 3 profesor, 4 doktor, dan 2 dosen yang sedang menempuh studi doktoral di UGM dan IPB University. Kompetensi dosen didukung rekam jejak penelitian, pengabdian, dan publikasi ilmiah yang sangat baik. Dalam 3 tahun terakhir, dosen PSPT menerbitkan lebih dari 220 artikel ilmiah, hampir setengahnya di jurnal internasional bereputasi. Dukungan sumber daya manusia yang berkualitas juga tercermin pada rata-rata beban kerja pengajaran, penelitian, dan pengabdian kepada masyarakat yang melebihi standar nasional. Penelitian dan pengabdian dosen PSPT tidak hanya menghasilkan publikasi, tetapi juga luaran nyata seperti pendaftaran varietas tanaman baru dan buku ilmiah. Kemahasiswaan PSPT menunjukkan capaian yang membanggakan. Pada tahun 2023, terdapat 112 calon mahasiswa yang mendaftar dan 50 orang diterima setelah seleksi ketat. Hingga semester genap 2023 hingga 2024, jumlah mahasiswa aktif mencapai 194 orang. Masa studi rata-rata mahasiswa antara 4 hingga 4,88 tahun, dengan presentasi kelulusan tepat waktu yang melampaui rata-rata fakultas. Sejak berdiri hingga akhir 2023, PSPT telah meluluskan 89 orang lulusan dengan rata-rata IPK di atas 3,3. Sebagian besar lulusan bekerja pada instansi pemerintah, lembaga riset, perkebunan swasta, dan sektor perbankan. Bahkan, banyak yang menjadi wirausaha mandiri. Beberapa lulusan juga melanjutkan studi magister, baik di dalam negeri maupun di luar negeri, seperti National Chongqing University Taiwan. Dalam mendukung proses pembelajaran, PSPT memiliki fasilitas yang lengkap dan representatif, ruang kuliah modern, ruang kerja dosen, laboratorium riset dan praktikum, hingga Museum Serangga Kutub Putih yang menjadi satu-satunya di Asia Tenggara. Museum ini telah menjadi pusat rujukan peneliti dalam dan luar negeri untuk sabbatical leave, magang, dan analisis spesies. Laboratorium PSPT terdiri dari lab taksonomi serangga, lab nematologi, lab fitopatologi, lab pengendalian hayati, serta lab biologi molekuler yang mendukung riset hingga level genetik. PSPT juga aktif menjalankan program Merdeka Belajar Kampus Merdeka atau MBKM. Mahasiswa berkesempatan mengikuti pertukaran dengan 12 perguruan tinggi lain di Indonesia yang tergabung dalam APSITA, mengikuti kampus mengajar, dan berbagai kegiatan pengayaan pengalaman belajar. Penelitian PSPT diarahkan pada pemanfaatan keanekaragaman hayati sumber daya lokal dalam pengendalian hama dan penyakit tanaman terpadu. Selama 3 tahun terakhir telah dilaksanakan 74 penelitian dosen yang diseminasi dalam puluhan seminar nasional dan internasional. Sementara dalam bidang pengabdian kepada masyarakat, PSPT memiliki 2 desa binaan dan puluhan program yang terintegrasi dengan kurikulum pembelajaran. Capaian lulusan PSPT terus meningkat. Sebagian besar alumni berhasil mendapatkan pekerjaan kurang dari 6 bulan setelah lulus, dengan tingkat kesesuaian bidang kerja yang semakin tinggi setiap bulannya. Berbagai keunggulan PSPT antara lain kualifikasi akademik dosen, kurikulum berbasis KKNI yang relevan dengan MBKM, publikasi ilmiah yang produktif, hingga fasilitas laboratorium dan museum yang mendukung pengembangan kompetensi mahasiswa. Sebagai program studi yang terus berkembang, PSPT Fakultas Pertanian Universitas Bengkulu berkomitmen menjadi pusat pengembangan ilmu proteksi tanaman tropis yang bereputasi internasional, menghasilkan lulusan yang kapabel, adaptif, dan siap berkontribusi dalam pengelolaan pertanian berkelanjutan berbasis sumber daya hayati.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Di ufuk barat Sumatera, di antara bentang alam yang menyimpan sejarah dan harapan, lahir sebuah ruang yang menumbuhkan mimpi menjadi nyata. Sebuah tempat di mana ilmu berkembang dan masa depan mulai dirancang. Inilah Universitas Bengkulu. Universitas Bengkulu adalah ruang bertemunya ilmu pengetahuan, inovasi, dan karakter. Di lingkungan yang asri dan dinamis, setiap langkah menjadi proses belajar, dan setiap interaksi menghadirkan kolaborasi. Disinilah generasi masa depan dipersiapkan untuk menjadi adaptif dan berdampak.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat datang di Universitas Bengkulu, tempat tumbuhnya ilmu, karakter, dan masa depan. Mari kita bersama-sama melangkah, berinovasi, dan memberikan kontribusi terbaik bagi institusi, bangsa, dan negara. Universitas Bengkulu unggul, berbudaya, berdaya saing internasional.
Sebagai universitas multidisiplin, Universitas Bengkulu menghadirkan beragam bidang keilmuan yang terus berkembang. Fakultas Keguruan dan Ilmu Pendidikan, Fakultas Hukum, Fakultas Ekonomi dan Bisnis, Fakultas Ilmu Sosial dan Ilmu Politik, Fakultas Pertanian, Fakultas Matematika dan Ilmu Pengetahuan Alam, Fakultas Teknik, Fakultas Kedokteran dan Ilmu Kesehatan. Setiap fakultas menghadirkan pengalaman belajar yang kontekstual dan aplikatif. Mahasiswa tidak hanya memahami teori, tetapi juga mengembangkan keterampilan melalui praktik di laboratorium, studio, klinik, dan lapangan. Proses pembelajaran mendorong lahirnya ide, inovasi, dan solusi nyata, menghubungkan ilmu dengan kebutuhan masyarakat, serta membuka ruang kolaborasi lintas disiplin. Kehidupan kampus menghadirkan pengalaman yang membentuk lebih dari sekedar akademik. Melalui organisasi, kegiatan seni, olahraga, dan komunitas, mahasiswa mengasah kepemimpinan, kreativitas, dan kepedulian sosial. Disinilah mereka tumbuh menjadi individu yang siap berkontribusi. Pengembangan ilmu pengetahuan diperkuat melalui riset dan pengabdian kepada masyarakat oleh Lembaga Penelitian dan Pengabdian kepada Masyarakat. Berbagai inovasi dihadirkan untuk menjawab kebutuhan nyata di lapangan. Kualitas pembelajaran juga dijaga melalui lembaga penjamin mutu dan pengembangan pembelajaran sebagai bagian dari upaya berkelanjutan membangun budaya mutu. Ekosistem akademik Universitas Bengkulu didukung oleh unit penunjang yang memperkuat layanan dan pembelajaran. Di antaranya, unit penunjang akademik Teknologi Informasi dan Komunikasi, Perpustakaan, Bahasa, Bimbingan konseling, serta pengembangan karir dan kewirausahaan. Hadir mendukung perkembangan mahasiswa secara menyeluruh. Didukung pula oleh pusat kajian yang mengembangkan riset dan inovasi di berbagai bidang strategis. Lingkungan belajar dirancang untuk nyaman, modern, dan inklusif. Ruang kelas representatif, konektivitas digital yang luas, fasilitas olahraga, serta akses ramah difabel mendukung aktivitas kampus yang produktif. Didukung pula oleh Rumah Sakit Universitas Bengkulu sebagai sarana layanan kesehatan dan pembelajaran, serta Masjid Baitul Hikmah sebagai pusat pembinaan nilai spiritual, melengkapi keseimbangan antara ilmu dan kehidupan. Berbagai capaian menjadi wujud komitmen terhadap kualitas dan tata kelola. Akreditasi unggul dari BAN PT, pengakuan internasional melalui akreditasi Aquin, serta predikat informatif dalam keterbukaan informasi publik, menegaskan posisi Universitas Bengkulu di tingkat nasional dan global. Universitas Bengkulu terus melangkah sebagai ruang lahirnya gagasan, penggerak inovasi, dan pusat kontribusi bagi masyarakat. Dari kampus biru di tepi barat Sumatera, untuk Indonesia, dan untuk masa depan. Universitas Bengkulu, conveying better future.
Kedamaian tercipta, ramah senyum menyapa. Buka mata dan rasakan keindahannya. Kedamaian persahabatan, cinta dan kebersamaan. Marilah semua kita tunjukkan pada dunia beragam pesona indah alam kita. Lestarikan bersama adat budaya kita, bergandeng tangan satukan langkah kita. Wonderful Bengkulu untuk Indonesia.
are the origin of a life harboring infinite potential. In the land of Rafflesia, we believe that agriculture is the DNA of innovation and the soul of sustainability. We are the innovation champion for sustainable solutions.
Our long journey began on April 24, 1982, driven by the spirit to advance the agricultural sector and contribute to the intellectual life of the nation. Faculty of Agriculture officially commenced its operation through the decree of the Minister of Education and Culture of the Republic of Indonesia. Today, for more than 4 decades, we have stood firm, shaped generations, and continued to transform ourselves toward excellence. The Faculty of Agriculture, University of Bengkulu has grown into one of the leading centers for education, research, and community service in the fields of tropical agriculture and coastal ecosystem management. Here, we do not merely produce graduates, we nurture innovators who are ready to make meaningful contributions to society and industry as a manifestation of the faculty's vision to become an excellent, cultured, and internationally competitive institution in the development of tropical agriculture and coastal ecosystem management by 2045.
Academic excellence is our utmost priority. At the Faculty of Agriculture, University of Bengkulu, we integrate the case method and project-based learning to cultivate and adapt generation equipped with practical expertise, critical thinking skills. To meet global challenges, we design a future-ready curriculum, becoming a vibrant home to over 3,100 students with proudly over 12 undergraduate programs and 5 postgraduate programs. Our undergraduate programs encompass agribusiness, agroecotechnology, soil science, animal husbandry, forestry, marine science, agro-industrial technology, plant protection, animal nutrition and feed technology, environmental science, agricultural extension, and fisheries science. We advance higher education through our postgraduate programs, including Masters in Agribusiness, Master in Agroecotechnology, Masters in Natural Resources Management, as well as doctoral programs in Agricultural Sciences and Doctor Nature Resources Management. Through a collaboration learning concept, we break boundaries, empowering students to explore knowledge both inside and outside the classroom. Together with the Faculty of Agriculture, University of Bengkulu, let us shape a sustainable future for agriculture and the environment.
Our grand vision is driven by the dedication of 183 professional faculty members. This academic strength is ensured by 38 professors, 33 associate professors, 47 assistant professors, and 40 instructors who are deeply committed to excellence. To foster a vibrant academic atmosphere, we provide an integrated learning ecosystem across 11 main buildings. This encompasses modern academic administration centers, advanced laboratories, shared lecture halls, and a comprehensive library. Our commitment to progress is continuous. We constantly invest in staff development and state-of-the-art facility upgrades. Through specialized sporting facilities such as our greenhouses and experimental fields, we ensure that theories taught in classrooms are successfully transformed into real-world solutions.
The Faculty of Agriculture fully supports students in developing research, academic and non-academic abilities with 11 student association and 3 student activity unit, as well as the Student Representative Council and the Student Executive Board, which support the development of study creative in organization, entrepreneurship, art and sport, which have result in national and international achievement. Student achievement in higher education extend far beyond a high grade point average. It encompasses all academic, non-academic, and character development accomplishment that shape students into competent, competitive graduates who are ready to face global challenges. For educational institutions, facilitating and according student achievement in a long-term investment that determine the university reputation, the quality of its graduate, and its tangible contribution to society. A strong alumni network and global partner ensure our graduates are prepared for the workforce. Alumni play a vital role in higher education. They serve as ambassador for their university and make substantial contribution to campus development. This role further strong connecting and strong relationship between alumni and the academic community, thus furthering campus growth and improvement. We collaborate with government and international institutions to create sustainable impact. Through this collaboration, various higher education institutions can share knowledge, experience, and resources to achieve the shared goal to producing quality graduates and contributing to community and national development.
We offer quality education in a variety of undergraduate and postgraduate programs, striving to the best.
Seeds are the origin of a life, harboring infinite potential. In the land of Rafflesia, we believe that agriculture is the DNA of innovation and the soul of sustainability. We are the innovation champion for sustainable solutions.
Our long journey began on April 24, 1982, driven by the spirit to advance the agricultural sector and contribute to the intellectual life of the nation. Faculty of Agriculture officially commenced its operation through the decree of the Minister of Education and Culture of the Republic of Indonesia. Today, for more than 4 decades, we have stood firm, shaped generations, and continued to transform ourselves toward excellence. The Faculty of Agriculture, University of Bengkulu has grown into one of the leading centers for education, research, and community service in the fields of tropical agriculture and coastal ecosystem management. Here, we do not merely produce graduates, we nurture innovators who are ready to make meaningful contributions to society and industry as a manifestation of the faculty's vision to become an excellent, cultured, and internationally competitive institution in the development of tropical agriculture and coastal ecosystem management by 2045.
Academic excellence is our utmost priority. At the Faculty of Agriculture, University of Bengkulu, we integrate the case method and project-based learning to cultivate and adapt generation equipped with practical expertise, critical thinking skills. To meet global challenges, we design a future-ready curriculum, becoming a vibrant home to over 3,100 students, we proudly over 12 undergraduate programs and 5 postgraduate programs. Our undergraduate programs encompass agribusiness, agroecotechnology, soil science, animal husbandry, forestry, marine science, agro-industrial technology, plant protection, animal nutrition and feed technology, environmental science, agricultural extension, and fisheries science. We also advance higher education through our postgraduate programs, including Masters in Agribusiness, Master in Agroecotechnology, Masters in Natural Resources Management, as well as doctoral programs in Agricultural Sciences and Doctor Nature Resources Management. Through a collaboration learning concept, we break boundaries, empowering students to explore knowledge both inside and outside the classroom. Together with the Faculty Agriculture, University of Bengkulu, let us shape a sustainable future for agriculture and the environment.
Our grand vision is driven by the dedication of 183 professional faculty members. This academic strength is ensured by 38 professors, 33 associate professors, 47 assistant professors, and 40 instructors who are deeply committed to excellence. To foster a vibrant academic atmosphere, we provide an integrated learning ecosystem across 11 main buildings. This encompasses modern academic administration centers, advanced laboratories, shared lecture halls, and a comprehensive library. Our commitment to progress is continuous. We constantly invest in staff development and state-of-the-art facility upgrades. Through specialized sporting facilities such as our greenhouses and experimental fields, we ensure that theories taught in classrooms are successfully transformed into real-world solutions.
The Faculty of Agriculture fully supports students in developing research, academic and non-academic abilities with 11 student associations and 3 student activity units, as well as the Student Representative Council and the Student Executive Board, which support the development of study creative in organization, entrepreneurship, art and sport, which have result in national and international achievement. Student achievement in higher education extend far beyond a high grade point average. It encompasses all academic, non-academic, and character development accomplishment that shape students into competent, competitive graduates who are ready to face global challenges. For educational institutions, facilitating and according student achievement in a long-term investment that determine the university reputation, the quality of its graduate, and its tangible contribution to society. A strong alumni network and global partner ensure our graduates are prepared for the workforce. Alumni play a vital role in higher education. They serve as ambassador for their university and make substantial contribution to campus development. This role further strong connecting and strong relationship between alumni and the academic community, thus furthering campus growth and improvement. We collaborate with government and international institutions to create sustainable impact. Through this collaboration, various higher education institutions can share knowledge, experience, and resources to achieve the shared goal to producing quality graduates and contributing to community and national development.
We offer quality education in a variety of undergraduate and postgraduate programs, striving to the best.
Seeds are the origin of a life, harboring infinite potential. In the land of Rafflesia, we believe that agriculture is the DNA of innovation and the soul of sustainability. We are the innovation champion for sustainable solutions.
Our long journey began on April 24, 1982, driven by the spirit to advance the agricultural sector and contribute to the intellectual life of the nation. Faculty of Agriculture officially commenced its operation through the decree of the Minister of Education and Culture of the Republic of Indonesia. Today, for more than 4 decades, we have stood firm, shaped generations, and continued to transform ourselves toward excellence. The Faculty of Agriculture, University of Bengkulu has grown into one of the leading centers for education, research, and community service in the fields of tropical agriculture and coastal ecosystem management. Here, we do not merely produce graduates, we nurture innovators who are ready to make meaningful contributions to society and industry as a manifestation of the faculty's vision to become an excellent, cultured, and internationally competitive institution in the development of tropical agriculture and coastal ecosystem management by 2045.
Academic excellence is our utmost priority. At the Faculty of Agriculture, University of Bengkulu, we integrate the case method and project-based learning to cultivate and adapt a generation equipped with practical expertise, critical thinking skills. To meet global challenges, we design a future-ready curriculum, becoming a vibrant home to over 3,100 students with proudly over 12 undergraduate programs and 5 postgraduate programs. Our undergraduate programs encompass agribusiness, agroecotechnology, soil science, animal husbandry, forestry, marine science, agro-industrial technology, plant protection, animal nutrition and feed technology, environmental science, agricultural extension, and fisheries science. We will advance higher education through our postgraduate programs, including Masters in Agribusiness, Master in Agroecotechnology, Masters in Natural Resources Management, as well as doctoral programs in Agricultural Sciences and Doctor Nature Resources Management. Through a collaboration learning concept, we break boundaries, empowering students to explore knowledge both inside and outside the classroom. Together with the Faculty of Agriculture, University of Bengkulu, let us shape a sustainable future for agriculture and the environment.
Our grand vision is driven by the dedication of 183 professional faculty members. This academic strength is ensured by 38 professors, 33 associate professors, 47 assistant professors, and 40 instructors who are deeply committed to excellence. To foster a vibrant academic atmosphere, we provide an integrated learning ecosystem across 11 main buildings. This encompasses modern academic administration centers, advanced laboratories, cell lecture halls, and a comprehensive library. Our commitment to progress is continuous. We constantly invest in staff development, and state-of-the-art facility upgrades. Through specialized sporting facilities such as our greenhouses and experimental fields, we ensure that theories taught in classrooms are successfully transformed into real-world solutions.
The Faculty of Agriculture fully supports students in developing research, academic and non-academic abilities with 11 student association and 3 student activity unit, as well as the Student Representative Council and the Student Executive Board, which support the development of study creative in organization, entrepreneurship, art and sport, which have result in national and international achievement. Student achievement in higher education extend far beyond a high grade point average. It encompasses all academic, non-academic, and character development accomplishment that shape students into competent, competitive graduates who are ready to face global challenges. For educational institutions, facilitating and according student achievement in a long-term investment that determine the university reputation, the quality of its graduate, and its tangible contribution to society. A strong alumni network and global partner ensure our graduates are prepared for the workforce. Alumni play a vital role in higher education. They serve as ambassador for their university and make substantial contribution to campus development. This role further strong connecting and strong relationship between alumni and the academic community, thus furthering campus growth and improvement. We collaborate with government and international institutions to create sustainable impact. Through this collaboration, various higher education institutions can share knowledge, experience, and resources to achieve the shared goal to producing quality graduates and contributing to community and national development.
We offer quality education in a variety of
tuber volume and weight. Conclusion and recommendation. In conclusion, planting media heights from 20 to 60 centimeters did not significantly affect pest damage or tuber components. Stem damage and large abundance were not significantly related to tuber yield components. However, tuber damage was positively and significantly correlated with tuber weight, but not with tuber length or diameter. Tuber weight also showed strong positive relationship with tuber length and diameter. For future studies, pest present and damage should be monitored periodically throughout plant growth rather than only at harvest. That concluded my presentation. Thank you for attention, everyone.
Okay, thank you very much for Bu Sri Upa. Next, we go to Bu Ella Nadia Erlindasan. Okay, Bu Ella. Hello. Go to Bu Ella Nadia.
Okay,
Okay, you can share by self your slide.
Yes.
You can share by self your slide.
Can you see my share screen?
Okay, good screen. Yeah, you can start now.
Good afternoon, all participants. First of all, I would like to introduce myself. My name is Ella Nadia Nasari. I'm from Universitas Pembangunan Nasional Veteran East Java. Today, I would like to present my research. The title is The Effect of Pruning Types and NPK Fertilizer Dose on the Growth of Curie. So, this background research as Curie is a horticultural commodity with the good market potential, particularly in the culinary industry. However, the productivity Curie in Indonesia is still relatively low compared with the market demand. One approach to improve Curie growth is through proper canopy management, such pruning. Pruning can reduce apical dominance and improve assimilate distribution toward productive organ. In addition, NPK fertilizer provide essential nutrients that support plant development, including reproductive growth. Although pruning and NPK fertilizer have been widely studied, their combination effect under different pruning types and NPK doses on Curie still require further investigation. So the study is to determine the effect of pruning types and NPK fertilizer dose and their interaction on Curie growth. The key question is which combination of pruning types and NPK fertilizer dosage provide the best growth response in Curie. So this research method was conducted from May to June 2026 at Mala Regency, East Java. The experimental design used a two-factor randomized block design. Pruning treatment is no pruning, main shoot pruning, pruning of the main shoot and lateral branch tips, and pruning water sprout. NPK fertilizer rate is 160, 320, 480, and 640 kg per hectare. All treatment combination were planting, the no planting treatment consistency showed the highest average number of flowers with 16.6 for leaf at 28 day, 38.9 for leaf at 35 day, and 50.5 leaf at 42 days. Meanwhile, the NPK doses did not show a significant effect during 28 until 42 days after planting. Although NPK dose 640 show a relatively higher growth than other doses. Next, I will explain the result for productive branches and number of flowers. For productive branches, which was pruning of the main shoot and lateral branches, produced the highest number of productive branches. The average value were 3.6 branches at 35 days after planting and 3.78 branches at 42 days after planting. This value were significantly higher than pit tree or water sport pruning, which produced only 2.11 and 2.36 branches at 35 and 42 days after planting. For NPK fertilizer N4 at 640 kg per hectare produced the highest number.
Do you have the picture? No?
Okay, okay, Mister. And now we continue with Dr. Nubailis. Okay.
Iya,
Okay, Mister. And now
Nubailis saja dulu.
continue with yeah. Okay, Mister. Dr. Nubailis.
Miss Midis. Okay.
Okay, okay, ma'am.
Yes.
Sorry, ma'am.
Who share the presentation? I have sent to the link before.
Okay, okay, ma'am. I will share you PPT.
Okay. Action. The problem?
Yes, ma'am.
No, not so.
Can you?
Okay. Okay, Assalamualaikum warahmatullahi wabarakatuh. For the first time, I would like to special thanks to the moderator who have given me a time to deliver my research with the title Potential of Filtrate from Several Trichoderma aspergillum isolate for Controlling Fusarium wilt disease on chili peppers. Next. Okay, next. The background of my research before the slide before. The background of my research: chili peppers, one of the horticultural plant that is very important, especially in West Sumatra. We know the production of chili pepper in Indonesia is still low when we compare with the optimal production of chili pepper, about 12 until 20 tons per hectare. There are many causing how the chili pepper production is low. But I want to focus the cause of plant pathogens. And also many plant pathogens can attack the chili pepper. And now I specialize to talk about Fusarium oxysporum, cause the Fusarium wilt disease. Usually in the field, the farmer control this disease by using the pesticide. We know that the pesticide dangerous for the environment, so we look for the alternative control by using biopesticide from the secondary metabolites from the antagonistic fungi Trichoderma aspergillum. Next. The goal of this research to obtain the best Trichoderma aspergillum isolate that producing secondary metabolite for controlling Fusarium wilt disease on chili peppers. Next. In this research, we use a randomized block design consists of 5 treatment and 8 replication. The treatment are the filtrate from Trichoderma aspergillum with the code A116, and the B treatment Trichoderma aspergillum with ACE-D 324 isolate, and then Trichoderma aspergillum with the code PP4, and then there is also negative control and positive control, and then the data we analyze by the statistic 8, and we continue with the analysis for LSD. Next. Implementation of the research in the laboratory. First, isolation of the pathogen Fusarium oxysporum, and then rejuvenation of Trichoderma aspergillum culture, and the isolate come from collection of laboratory, and then preparation of the filtrate from the Trichoderma aspergillum isolate. Next. And then we continue with the implementation of the research in the greenhouse. In the greenhouse, we did preparation of planting media And then the media we treat with the pathogen Fusarium oxysporum. And then before we make the seedling, the seed we treat with the filtrate of Trichoderma aspergillum. And then after seedling want it to move to the greenhouse, the root of the seedling we treat again with the filtrate of Trichoderma aspergillum. And then we continue to planting with the pots, and then we maintenance and harvesting. Next. The recent observation of the parameter observed, Fusarium wilt, first the Fusarium wilt disease, there is the incubation period, disease incident, disease severity. And then the plant growth, there is the plant height, number of leaves, appearance of the first flower, and fruit weight. Next. Result and discussion. The incubation period in the table 3 we can see and the treatment from filtrate of Trichoderma aspergillum with the code A116, only two plants infected, and the control there is a 6 plant infected. And the symptom symptom of eating start from 22 to 33 days after inoculation. Treatment with filtrate Pomperius Trichoderma aspergillum isolate prolong the incubation period of Fusarium wilt disease. Next. After that, we continue with the disease occurrence. This I saw with the stem graphic. We can see at the graphic the filtrate from Trichoderma aspergillum with the code 116. There is the activity the higher than isolate, and the other isolate. The higher the activity to inhibit to reduce the disease occurrence 66%, and also disease severity so that the filtrate from Trichoderma aspergillum with the code 8116 is reduce the disease severity higher than the other filtrate. Okay. Next. And this is the growth of the plant, vegetative states and generative states. We also get that the filtrate from Trichoderma aspergillum with the code 116 is better than the other isolate. And also the generative state, the filtrate from isolate A116 accelerated flowering to 42.50 days, and higher the effect at 12.335. Next. The conclusion of the research result indicate that treatment with filtrate from Trichoderma aspergillum isolate show potential for suppressing the development of fusarium wilt and enhancing chili plant growth. The Trichoderma asperellum with A116 isolate filtrate proved to be the most effective isolate with the effectivity 66.66% in reducing disease incidence and 79.12% in reducing disease severity. Furthermore, the Trichoderma asperellum A116 isolate filtrate increase the chili plant height by 27.10% and boosts chili production, resulting in 119.37 increase in food weight. Next. Thank you. That's all. Thank you for your attention.
Okay, thank you, ma'am, for your presentation. Before we go to next presenter, we are now discussion, and for all participants in this room, please raise your hand and give your question. Okay, I will ask to Ma'am Nurbailis.
Yeah.
Ma'am, what the formulation you use for application the metabolite to
Okay.
control the fusarium?
Okay. Directly answer? Or you want to collect the other questioner?
Yeah, and the question for
Directly.
Ma'am Nurbailis?
Okay. Okay.
Yes, directly, ma'am.
Okay. I want to answer your question. What formula? Is it the what we say culture cair? Yeah. We from the what we say culture cair. We make iso the filtrate mean after we met the culture cair, and then we'll filter. Yeah, we'll filter with the what we say the, and then in the filter the mass of trichoderma, and we assume that the filtrate contain also secondary metabolite of Trichoderma asperellum.
Okay, ma'am. Okay. You not use another material add to the formulation, yeah? Only use the culture filtrate.
Yeah. Yes. Only
Okay, you
use the
have-
culture filtrate. Not isolation with
Yes.
the
You have application in the field?
no, only the at greenhouse.
Oh, greenhouse, yeah, still
Yeah,
in greenhouse.
Yeah.
Okay, ma'am, thank you.
Yeah. Welcome.
Any questions
Recording stopped.
for Ma'am Nurbailis? Oh, in the chat room, any question? For Ma'am Nurbailis, what is the main secondary metabolite in Trichoderma that play a role in controlling fusarium wilt in Chile?
Okay. Yeah, we assume that there is the compound, a chemical compound in metabolite that function as the
Recording in
antimicroba.
progress.
Yeah, like can inhibit the growth of fungi. Like the I forget the name of the compound.
In Indonesia, what in Indonesia? Can
No,
Indonesia, ma'am.
the name of the chemical compound, like flavonoid, for the general flavonoid, but in the member of flavonoid, there is the compound as a function as the antimicrobial. Okay. There is another question.
Okay. How do you think, Miss Mr. Nurfianto, about the answer? Hello, Mister. Okay. We continue with the next question for Mr. Sang.
Recording stopped.
From Ibu Silpi Ikawati. What is next project related you research that you present in this seminar?
Yeah. Thank you, moderator, and also thank you for Ibu Sylvie Ikawati, and happy to see on our, yeah, and also happy to see on our presenter again. Thank you for your really impressive question from Ibu Sylvie Ikawati about what is the next project related to my research. After I did the first step is determine which is the dominant fruit-fly species on the curly chili in West Sumatra. Of course, the next step we have to go to find the method to control or prevent the wood fly pest. For example, follow the mechanical. For mechanical, of course, we can find because spot West Sumatra, they have a lot of chili varieties, over 30 varieties. So we can among 30. So we can find which one get the highest resistant potential to the wood fly infestation. So according to the mechanical. Similarly, we also based on the chemical. Of course, we can find to find out which chemical compound can give the effective control on the fruit fly Apterina orientalis. And also based on in some of the biological controls, we also can find the bio insecticide or biofungicide to control. So this is the follow-up research I gonna to do after my study findings. Thank you. That's all.
Okay, thank you for your answer the question. Still any question? I will ask to Ma'am Shahrawati. Hello, ma'am.
Yes, I'm here.
Yeah. How long life cycle of the Pardosa, ma'am?
We never did the research about the life cycle of Pardosa pseudoanolata at laboratory, but in references, some researcher already reported that Pardosa pseudoanolata have the long life cycle, particularly when they reach the adult, often the adults. So maybe I can give you information, the data about laboratory.
Okay, ma'am. Okay. We go to continue with the next
Recording in
question.
progress.
And now to Mr. Irfandri. Time for use.
I send to my PPT to committee. Yeah.
Yes, Mister.
Yeah, I send to my PPT, the committee, organization committee.
Okay, okay.
Yeah,
Okay, Mister. I
Yeah.
will help you to share PPT.
yeah. Irfandri. Irfandri.
Mister.
Yeah.
Sorry. You send your PPT?
Yeah. I send to my PPT and committee.
Okay, Mister. Sorry, I will looking for you PPT and for this time I change ya. Now, times for Ibu Jurai Resti.
Recording in progress.
Data set contain 4,957 annotated image. We use from RoboFlow available, publicity available, and there is 10,561 bounding box for each class. On this slide, we can see the data distribution for each class pets. There are 4 class pets. There are Benicia tabasi, 1,722, and Caterpillar 2,505, and Mazes Pertisae 3,000 589, and trips class or trips Pests 2,740. We split the data into train, validation, and testing. But the same experiment setting to conduct our experiment. Okay, yeah. Next slide. Yeah, there are three model yellow elephant. There are yellow elephant nano or yellow elephant N, and yellow elephant small or yellow elephant S, and yellow elephant medium or yellow elephant M. Each model have a different parameter number, but we conduct our experiment in the same setting, like input size 640 by 640, and same split, and then optimizer Adam W, and the same early sorting. Okay, next slide. In our study, the result of the experiment, we obtained that yellow elephant N has the best performance. We can see on the slide that yellow elephant N has 0.700 accurate than the other models, and the same too for precision. Yellow elephant nano has 0.675 more higher than for the other models. But we can see for yellow elephant M or yellow elephant medium has a recall value more higher than the other models. This because for this model, yellow elephant medium, it's have the big architecture and the big parameter number. Okay, next. Yeah. Yeah. Same for before slide, that yellow elephant N or yellow elephant nano has the best accuracy and training efficiency. For the training efficiency, yellow elephant nano has 0.881 hours. The time run computational more lowest than for each model. For each model. Okay, the next slide. Okay. On the slide, on this slide, we can see a confusion analysis map. This slide show that for diagonal value for class C or class caterpillar class have 0.94 accurate, and for class T or trips has 0.99 accurate. But in contrast with BT class, just has 0.71, and MP class just has 0.81 accurate. And we also can see that for BT class has 0.21 or 21%. The data or the object of BT class detect terdeteksi or detection in false or negative class. It is MP class. For C class has a confidence 0.94 and T class 0.99, and for MP class has 0.81 and BT class has 0.71. Okay, next slide. On this slide, we can see the example for data image for class for caterpillar class. We can see like image number 1, 2, 3, 4 row, first row, column 4. C class have 0.8 confidence, and we on the slide we also can see that BT class has confidence 0.3. It means that the background image is detect as BT class. Jadi background image is detect to BT class with confidence 0.3. Okay, next slide. Okay, the final in our study conclusion, YOLO 11N or YOLO 11 Nano provide the stronger accuracy, efficiency balance for this CV Pest dataset with accuracy 0.700 and lowest training cost 0.881 hours, and for BT and MP class has a lowest accurate. For this result suggests that for the future improvement we can reduce class confuse and reduce background error. That's all. I think. Thank you for the attention, and wassalamualaikum warahmatullahi wabarakatuh.
Puryanto. Halo, Bapak
Ya,
Puryanto.
ya siapa?
Oke, silakan, Bapak. Puryanto. Bapak.
Ya, ya siapa? Sudah terlihat ya Pak? Itu saya. Sudah terlihat ya Pak?
Belum, Pak.
Ya, ya siapa? Sudah terlihat ya Pak? Sudah terlihat Pak?
Oke, sudah. Silakan di slideshow, Pak.
Right, good afternoon, all participants. First of all, I would like to express my sincere gratitude for the opportunity to present our research at the second National Seminar on Plant Protection 2026. My name is Puryantoro from the University of Abdurrahman Saleh Bondo. Today, I am pleased to present our study in paper Determinant of the Intensity of Climate Change among Smallholder Arabica coffee farmer, evident from an ordered logit model in Indonesia. The study was conducted together with Johnny Multimulya Aji from the University of Jember and Swiss Tendency from the University of Abdurrahman Saleh Bondo, East Java. Our study focused on understanding not only whether farmer adapts to climate change, but more importantly, how intensively they implement adaptation strategies. Let me begin with the background of the study. Climate change has become an important challenge for agricultural production, including Arabica coffee farming. Changes in temperature, rainfall pattern, and the increasing aggression of extreme climate event can affect coffee productivity, product quality, and the long-term sustainability of coffee farming. For smallholder farmer, these changing conditions create increasing uncertainty. Therefore, adaptation strategies are essential to strengthen the resilience of coffee farming system. However, there is an important research gap. Previous studies have predominantly treated climate adaptation as a by decision, meaning that farmers are simply classified as either adapting or not adapting adaptation practices. But this approach does not show how intensively farmers are adapting. For example, one farmer may implement only few adaptation strategies, while another farmer may implement a more comprehensive set of strategies. Therefore, empirical evidence regarding different level of adaptation intensity, particularly among smallholder or Arabica coffee farmer in Indonesia, remain limited. We study undersized that gap by implementing an ordered logit model to examine different level of climate change intensity. Based on this background, the study has two main objectives. First, to classify the intensity of climate change adaptation among smallholder Arabica coffee farmer. Second, to identify the socio-economic determinant that influence different level of adaptation intensity by using an ordered logit model. In other words, we wanted to answer two main questions: how intensively are smallholder coffee farming adapting to climate change, and what factor influence their ability to likelihood to achieve higher level of adaptation. Next, I will be briefly explain the research method. The study was conducted in the Mount Ijen Helen area in East Java, Indonesia. Specifically, this research covered Sumberwaringin District in Bondowoso Regency and Arjasa District in Situbondo Regency. The study involved a total of 146 smallholder Arabica coffee farmer. Among them, 37 farmer were from Bondowoso and 49 farmer are from Situbondo. These respondents were selected using simple random sampling. To measure adaptation intensity, we assess 5 climate adaptation strategies. First, agroforestry practices. Second, the use of climate resilient coffee varieties, and third, organic fertilizer application for mulching, and fifth, access to climate information. Based on the number of strategies implemented, farmer were classified into 3 categories. Farmer implementing the 3 strategies were classified as having low adaptation intensity. Those implementing 4 strategies were classified as moderate, and those implementing all 5 strategies were classified as having high adaptation intensity. To identify the determinant of adaptation intensity, we applied on ordered logic model using 8 explanatory variables, including field size, education, farming experience, plant age, farmer group membership, access to credit, and non-farm activities. Now, let us move to the main finding of the study. The first important finding is that climate change adaptation among smallholder Arabica coffee farmers is predominantly at the moderate level. A total of 30.8% of farmers implemented for adaptation strategies. Meanwhile, 14.4% were classified as having low adaptation intensity, and only 4.8% achieved high adaptation intensity. This finding indicate that adaptation is already taking place among coffee farmers. However, comprehensive adaptation remains limited. In other words, most farmers have started to adapt, but only a small proportion have implemented all available adaptation strategies. This most significant determinant is farmer age. The results show that farmer age has a negative and statistically significant effect on adaptation intensity. The analysis indicates that older farmers are less likely to reach higher level of adaptation. More specifically, its additional year of age decrease the likelihood of being in higher adaptation category by approximately 4.7%. One possible explanation is that younger farmers tend to be more open to new information, new technologies, and changes in farming practices. They may also be more willing to experiment with new approaches to respond to climate-related challenges. Therefore, this finding highlights the importance of encouraging younger generalists to become involved in coffee farming. The participation may contribute not only to the sustainability of the coffee sector, but also to the development of more innovative and climate resilient farming system. The second significant determinant is access to credit. Our findings show that financial access plays an important role in supporting higher adaptation intensity. Limited access to financial resources can become a barrier to more intensive climate adaptation. This is understandable because many adaptation practices require initial investment. For example, farmer may need financial resources to invest invest in climate resilient coffee varieties, develop agroforestry system, apply organic management, or implement soil and moisture conversion practices. Therefore, adaptation is not only a matter of knowledge or willingness. Farmer also need sufficient financial capacity to transform their adaptation plans into actual farming practices. This finding emphasizes the importance of improving inclusive agricultural financing for smallholder coffee farmers. To conclude, there are several key findings from the study. First, climate change adaptation among smallholder Arabica coffee farmers is predominantly and at moderate level. Around 0.8% of farmers implemented for adaptation strategies, while only 4.8% risk high level of adaptation intensity. Second, the audit logic analysis identified farmer aid and access to credit as the significant determinant of adaptation intensity. Farmer aid has a negative effect, indicating that younger farmers are more likely to achieve higher level of adaptation. Meanwhile, access to financial resources is essential because it enables farmers to invest in more comprehensive adaptation practices. Therefore, strengthening climate resilience among smallholder coffee farmers requires an integrated approach. This should include inclusive agricultural variety, effective extension service, access to climate-related information, and greater participation of younger generations in coffee farming. In summary, strengthening climate resilience requires not only adaptation technologies but also the financial capacity and human capacity to adopt and sustain those changes. Thank you very much for your attention. Not only adaptation technologies but also the financial capacity and human capacity to adopt and sustain those changes. Thank you very much for your attention.
President, you on the room? Jamila, apakah ada Miss M? Oke, baik, terima kasih Pak Puryanto atas presentasinya. Next presenter Bapak Muhammad Mustafa. Thank you very much. My clients. Sudah, Pak. Sudah, tapi suaranya agak ini Pak, putus-putus Pak. Clear. Kalau PPT-nya sudah terlihat, tapi suara Bapak yang putus-putus. Clear. Sudah kedengaran? Oke, baik. Memang bisa menggunakan delivery
my article by the title Tilapia Supply Chain Export Bengkulu. We have to spotlight the investors about the tilapia. Totally, Bengkulu contribution was about 1.22% of national production of tilapia. The central production in the North Bengkulu, but really got that source of one of the biggest contributors to the national production and also to the production of tilapia in Bengkulu province. That's why we choose Source Bengkulu to identification with the extraction of supply chain and also their production, and also identification the form of production and financial flows in the supply chain system. Okay, that's our existing research scale. Research specifically on the tilapia supply chains, the mainly in the Bengkulu province, particularly regarding comprehensive mapping in Bengkulu province. There are any research study, study and other Bengkulu study. They are focused on agriculture community, not in the poultry and also in the fish. So that's why we focus on research on the specific partner, each solution should be disease and better characteristic of the supply chain, these regions, politically, financial, based on then we primarily is identifying politic profit using the hands chain efficiency and then this after optimize the now segments used in the concept we divide three. First one is identification of and then mark material there. The supply chain. The current of responded we divide it into two farmers and customer, and then in farmers there we try education the use and economic of the chains. And the last one is supply chain. Application for information flow, finance flow. Before it, we strive flows product up and the this is about finding education of our students to answer objectives of the vista. This is to product informations also patterns. This pictures actually describe those finance and product. From farmers to consumer, wide span of supply chain channel. Each channel is contribution to the profit farmer, also contribution to the consumer to the landscape of distribution. And then the findings and findings of supply chain sector, there are for what pipeline, this is actor, emmer, collector, wholesaler, retailer, and consumers. From the pictures, we can put each of the channel. It has less close have actors. For instance, in channel 1, we only have 2 actors from farmers directly to the consumer. But channel 4, channel 5, we have 4 from collect agent, then the retailer, finally consumer. And also the five the most in each of the channels, the little profit or margin that we get by each of the channel. Now, because it's distributed better and it's each actor of each channel, we can describe from pool of creative product finance information. They are rules in specifically channel 1 to the channel 5. Then this is the distribution of profit market and farmers have how we can profit from each actor in each channel supply chain in more actors. Actually, the biggest profit can get by actor in supply chain channel compare actor in supply chain channel 5, they only 74 compared to the channel of 100% pounds. Okay, the conclusion that key benefits of the supply chain is farmer, collector, seller, and consumer. The flow of chain of product about 84.5 tons tilapia producers cycle. And then financial characterized by notice payment on one side, depending transaction of actor in the pipeline house. And the is a person flow, capacity, product quality, price, quality, timing, and transaction location. It's crucial in identifying the information flow from farmer and consumer or the person consumer. Okay, let's determine it's our expectation. Thank you very much, and sorry for inconvenience in being the president. Assalamualaikum.
Next chapter, Mesdan.
Assalamualaikum to all of you. Good afternoon. Can I start now? Assalamualaikum to all of you. Let me know when it's shared. Is it visible to all of you? Can I start now?
Yes.
Okay. Assalamualaikum to all of you. Let me know when it's shared. Is it visible to all of you? Can I start now?
Yes.
Okay. Assalamualaikum to all of you. First of all, I want to introduce myself. My name is Mehri Nayaz. Let me know when it's shared. Is it visible to all of you? Can I start now? It's slide share show. Okay. Assalamualaikum to all of you. First of all, I want to introduce myself. So first of all, I want to introduce myself. My name is Mehri Nayaz. I am a PhD scholar at the University of Bengkulu in the plant protection. It's like so basically today I will present my study on the title of Mealybug Pseudococcidae associated with the date palm. Myself, my name is Mehri Nayaz. Give me a second, please. Okay, mealybug associated with the date palm in Bengkulu province, and I have done this study under the supervision of Pagostin Zarkani. So start with the associated with the date palm. Start with the introduction. So first of all, let me tell you about the mealybug associated with the date palm. Basically the important self-sucking pest of many crop, including palm, and they have a wide range of host plant. They can attack any kind of host plant, and the date palm is economically very important fruit crop cultivated in tropical and subtropical, including Indonesia. If we talk about the infestation, so they can reduce the plant growth, fruit quality, and the productivity also. So you can conclude it that their effect is really worse for the crop. And in Bengkulu province, information about the mealybug associated with the date palm is still very limited. So we wanted to find out which mealybug species are actually associated with the date palm and how we differentiate them accurately. And so for the research to answer this question, we have three objectives. First, we conducted the field survey and collected the mealybug specimen from the date palm tree. Secondly, we identified the species on genus and species level, and we are using the morphological characteristic. And thirdly and finally, we use these characteristics to develop the clearer identification information for the species we found. So basically the study followed three steps: fieldwork, collection of the specimen, and thirdly identify the species. So my research gap is really clear and very accurate, that there is no such accurate data available regarding on the mealybug associated with the date palm, especially in Bengkulu province. Let's talk about the methodology. So in methodology, I did a survey, and I did survey to some places all over the Bengkulu province, which include Lebong, Rejang Lebong, Kepahyang, Saloma, Bengkulu Utara, and Bengkulu Tengah. And I have found the date palm trees in Lebong, Rejang Lebong, Kepahyang, Bengkulu Utara, and Moko. And these in this area, I collected around many species from specifically from the 8 locations. And in this table, you can easily see these are the exact location that I found the data of the mealybugs, and these are the plant hosts which shows the root, branch, stem, and the fruit part of the plant, which I collected the data. And I have collected many specimens. Here you can see the interesting part of my study is, after preparing and examining the specimen, we identify 4 different mealybug species. And as you can see here in these images, starting with the first image, we identify the Dismicoccus brevis. The second image shows Palmicultur palmarum. The third one shows Planococcus thaimensis, and the fourth one is Nymphococcus nympha. So apparently they are looking very similar, but we can differentiate them by their molecular characteristic, and I will show you in the next slide. The third one shows Planococcus thaimensis, and the fourth one is Nymphococcus nympha. So apparently they are looking very similar, but we can differentiate them by their molecular characteristic, and I will show you in the next slide. The third one shows Planococcus thaimensis, and the fourth one is Nymphococcus nympha. So apparently they are looking very similar, but we can differentiate them by their molecular characteristics. I will show you in the next slide. The third one is so apparently they are looking very similar, but
Recording in progress.
Halo, melihat? Sepertinya terpental ini. Mungkin
Recording
sambil
in
menunggu
progress.
beliau masuk lagi, kita akan melanjutkan sesi diskusi. Sepertinya terpental ini. Mungkin
Recording in progress.
Okay, thank you, ladies and gentlemen. We will now move on the second question and answer session. In the second session, will be open the opportunity to three participants to ask questions. For those who would like to ask question, please mention your name and institution. In the second session, will be open and opportunity, especially with presenter your question is addressed to. We will like to invite the first three participants to ask three questions. Silakan Bapak Ibu, ada yang mau bertanya dan nanti Bapak Ibu silakan pertanyaannya ditujukan ke siapa, untuk siapa seperti itu. Silakan Bapak Ibu, ada yang ingin bertanya, ditanyakan Bapak pada pemateri atau presenter pada hari ini ditujukan ke siapa. Sepertinya untuk presenter sudah semua. Silakan Bapak Ibu, ada yang ingin bertanya, ditanyakan cuma satu ya Pak. Rachel materi atau permainan pada apakah ada Bapak Rachel sah? Siapa? Sepertinya untuk presenter sudah semua. Silakan Bapak Ibu, ada yang ingin bertanya, ditanyakan cuma satu ya Pak. Silakan. Di sesi yang kedua ini ada kita Rachel sah? Siapa? Sepertinya untuk kita mulai untuk diskusi Bapak Ibu. Ada yang mau bertanya tentang tadi Bu Jamila, silakan terkait tentang nematoda. Di sesi yang kedua ini ada kita apa tadi tadi terkait tentang kita mulai untuk putih-putih Bapak Ibu di kurma seperti itu. Ada yang mau bertanya tentang tadi kopi Pak, silakan Budiantoro tadi dan ada terkait tentang apa tadi artificial intelligence terkait tentang AI putih Bapak Ibu di kurma seperti itu. Disampaikan oleh Bu Etnawati Budiantoro tentang gimana beda terkait tentang mendeteksi apa penyakit atau artificial intelligence. Silakan Bapak Ibu, ada Bu Reswita. Boleh, jadi boleh pakai bahasa Indonesia lah biar apa namanya lebih mempermudah kita kalau ini ya. Jadi sesi yang kedua ini.
I'm really sorry, I have interruption. I just changed my location so I'm able to speak. Can you please share my PPT?
Yes.
Okay, thank you.
Oke, kita lanjutkan dulu untuk Mas Har, kalau seandainya masih ingin disampaikan. Halo, Mahren, would you like to continue with presentation?
Deka, co-host.
I can't see the screen.
Ya, Ibu.
Deka, please help to show the PT Mehren Puput PT.
Baik, Bu.
Terima kasih, Pak. Next slide, after methodology. Slide number 8. 8. Next. Okay, this one. Thank you. Okay, so for this slide, after preparing and examining the specimen, we identify 4 millibug which are different with genus, belonging to different genus, and you can easily see them here in these images. Starting with the first, we identify Dismicoccus brevives. The secondly is Palmicultur palmarum. The third is identified by Planococcus thymamensis, and the fourth one is Nymphococcus Nifa. So apparently they are looking very similar, but their morphological characteristic make them different from each other. And this leads to the next question. Next, please. How did we know that they are different? So for that, we needed to look much closer to the morphological characteristic. Next, please. For the Dismicoccus brevives. Next slide, please. Can you please, next slide, please? Okay, for the Dismicoccus, can you can see in the image, this is actually the most frequently recorded species, the most dominant in my study. First, if you look at the overall body, it's looking more like oval to rounded in shape, and the important characteristic of this species they have 17 pair of cirrhi, the dot-like structure that you can see in the margin of their body, and they also have the wax filaments, which is like a hair-like structure in all over the body, and they also have pores and ducts. So we not identify the species just because of their shape, but there are many characteristics included that can help us to identify the species. Next, please. The second one is Palmicultur palmarum. In this, I will just go for the very highlighted characteristic. So you can see the conical structure, which is the seta, and the multilocular pores, they are present, which help us to identify this species. Next, please. Another species is Planococcus thymamensis, in which we can easily, next please. Okay, this one, you can easily see this, the number of cirrhi and multilocular pores are help us to identify this species. Next is the Nymphococcus nympha. Next, please. In which we have the conical structure, and they have a tubules which are present all over the body, which help us to identify this species. Next, please. Okay, so in this overall, as you see in the graph, the Dismicoccus brevives was the most frequently species, which represented 70% of the confirmed species, and which is followed by the other species, which are Palmicultur, Planococcus, and Nymphococcus nympha. So it means, based on our survey, Dismicoccus brevives was the most dominant species among the confirmed species. Next, please. So let's move towards the discussion. So another interesting finding was where the millibugs were present in our survey. So they were associated with the different parts of the plant of the date palm, including the roots part, the branch, stem, and the fruits. So for the further monitoring, it's really important to inspect the date palm more completely, including above the ground and below the ground. So it's really helpful for the future monitoring and for the future study. So thank you so much, and thank you for your attention. And I just concluded my all study. Can you please, next slide? I just want to conclude in my study. So we did survey all over the Bengkulu province, which I mentioned the location, and we found the 4 species which is belonging from the 4 different genera, and these really helpful for these millibugs are really helpful for the contribute to understand of the millibug complexes associated with the date palm in Indonesia, and provide a base for the future monitoring and best management study, and especially in Bengkulu. And thank you so much for your attention. I will be very happy to take any question.
Okay, thank you. Your question. Selamat sore semuanya Bapak Ibu. Tetap semangat. Apakah kita ada tanya jawab? Silakan. Kalau seandainya ada yang ingin ditanyakan, your question.
Oke, moderator.
Oke.
How to take sample from root? Are you asking this question? So it's
Recording
very
stopped.
visible. It was very visible, like we didn't dig the soil. It was present on the top of the root. We just need to remove some of the highlighted of the outer part of the roots. So we didn't dig the soil. So it was present very apparently. It was visible, and we collected with the brush, and we put in the 70% of alcohol, and the rest of the thing we done in lab. So it was visible to collect the sample. It was very easy.
Okay,
Yeah, thank you, Bu.
thank you.
Yeah.
Oke, thank you. Friend, ya. Bu Jamila, sudah? Apa ada yang mau ditanyakan lagi? Bu Hafizah?
Kalau yang apa tadi yang AI.
Oke, AI, ada Bu Ernawati.
Ya, apakah aplikasinya itu cuma untuk mayor pest chili?
Bu Ernawati, apakah masih bersama
Gak
kita?
ada orang yang masih itu, coba gak ada orang.
Halo, Bu Ernawati?
Iya, jadi mayor pest chili itu kan banyak ya.
Ya.
Kalau mungkin ada yang juga di tanaman yang lain gitu, jadi saya pengen tanya.
Sebenarnya menarik itu, Bu. Jadi ada
Menarik, tapi orangnya tidak ada.
Kayaknya orangnya kemana nih? Halo, Bu Ernawati? Apakah masih bersama kami?
Ernawati.
Oke, baik. Mungkin ada yang lain, Bu Hafizah? Ada yang ingin ditanyakan atau ingin diskusi? Kami persilakan.
Yes, kalau yang lain.
Kalau sepertinya Pak Mustafa. Oke, kalau tidak ada, kita sudahi saja dulu mungkin. Oke, ladies and gentlemen, before we left the room, we will invite everyone to turn on your camera. Jadi buka videonya untuk foto bersama. Please turn on your camera. Get ready for the photo. Deka, please.
Oke, baik. Mungkin izin saya memimpin Bapak ya untuk foto bersama. 1, 2, 3. Sekali lagi. 1, 2, 3. Udah, Bu.
Oke, thank you. Terima kasih semuanya Bapak Ibu yang sudah hadir pada kesempatan sore hari ini, dan kita akan mendengarkan siapa sih yang menjadi best presenter ya. Penilaian best presenter di International Seminar on Plant Protection Bengkulu University. Jatuh yang bagus adalah yang melalui penilaian dan jatuh pada instansinya adalah Universitas Jambi. Selamat kepada Hafizah Nahlunisaya. Selamat pada Bu Hafizah atas presenternya, dan terima kasih Bapak Ibu semuanya yang sudah hadir di kesempatan seminar internasional pada sore hari ini. Thank you very much. Mohon maaf apabila ada salah-salah dalam penyampaian maupun memandu acara pada sore hari ini. Kami akhiri. Bila taufik wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Baik,
wabarakatuh.
kita ketemu tahun depan lagi Bapak Ibu.
Insya Allah Pak.
Terima kasih.
Iya.
Ya.
Recording in
The
progress.
research with factorial complexity random design with factor 1 comparison of spent mushroom substrate LB and co-manure PT 100% substrate 90% mushroom backlog and 10% cow manure until P6 100% of cow manure. Factor 2, we mix streptomyces and no streptomyces. Each treatment unit we do in 5 plant polybag. We start the research. We start from multiplication of streptomyces in rice. And we make the streptomyces formula by mix inoculum with talc and calcium carbonate and milling to powder. And second, we continue to the composition of used mushroom backlog with formulation 550 kilo used mushroom backlog with cow manure, depend on composition, and 50 milliliter of molasses. And then we ferment it for 2 weeks. 2 weeks. And after that, we formulation of organic fertilizer with the compost of mushroom backlog with 50 gram streptomyces inoculum. And then we mix with soil to make the growing medium. And then before planting, we inoculate 10 milliliter of Erwinia suspension. And we incubation per week and planting 5 seeds per polybag. And then we maintain for 8 weeks. Okay, this the composition, the composition of H mushroom. And after observation, we found, most of the plant is healthy, and some of them has had disease, had disease like this. Okay, had disease like this. And then application of organic formula with streptomyces can increase the growth, can increase the growth, growth of like plant egg. This formula no streptomyces, and this formula with streptomyces, yeah, more than, yeah, per plant head, yeah, number of leaf, yeah, number of leaf, also number of leaf, and then reduce disease intensity, yeah. Formula with streptomyces can reduce the disease intensity around 30%, yeah. This until 40% disease in streptomyces formula, only 14%, yeah, 40% disease intensity. Okay, conclusion, the application of an organic fertilizer formula derived from spent mushroom extract and with streptomyces can enhance the growth and yield of shallot and reduce the severity of short rot disease. And the most effective organic fertilizer formula consists of a blend of spent mushroom substrate of the crop and livestock with ratio 80 and 20%. Yeah, we enrich with streptomyces. Okay, thank you very much.
Okay, thank you, Mr. for your presentation. And now we are discussion. And I see in chat room many questions. For Ibu Niken, could you please explain the size range of the marker used to visualize the PCR result? From what I can see, the PCR product size is between 200 and 300 base pairs. And to Ibu Jurai, based on the research finding, have plans been made for field application? Okay. Please you answer to Ibu Niken and Ibu Jurai.
Okay, thank you for Ibu Sampurna Ginting. I will answer the question. For the PCR confirmation, we use 100 bpdna liter to estimate the size of
How do you think, Ibu Mitfah? Okay, next to Ibu Jurai.
Yes, thank you for the question. It's a very interesting question. Yes, our research it's only in laboratory research. Yeah, it's in vitro test. So the future we will go to implanta research to apply or implication the filtrate to the plant. Maybe first we begin to the greenhouse scala, and then after that go to the field scala. Before application to the field, I think we must to make a formula to stabilize the filtrate and to make sure that the filtrate is ready to application to the field test. I think it's the answer. Thank you.
Okay, Ibu Jurai, thank you for your question. And now we are going to Ibu Tricelia. Have many questions in the chat room. The first question from
Bu Siti.
Yeah. Where was the Trichoderma aspergillum isolate, which function as entomopathogen, original of time from? What is the difference between this Trichoderma aspergillum and Trichoderma aspergillum that function as a biological control agent or beneficial fungus?
Thank you for Bu Sampurna. Trichoderma aspergillum that used in the research is endophytic from crops, several crops. Endophytic, not rhizosphere. Origin endophytic. This trichoderma before my research, I have isolated endophytic from crops, and I have trichoderma, and after identification by molecular, this species is Trichoderma aspergillum. So as we know, trichoderma usually control of plant pathogen, but in my research, trichoderma can use to control insect pests. But naturally, trichoderma is rare as entomopathogen. But from previous study, we can get trichoderma infection of the species of insect from natural. So in this research, we use endophytic isolate, really not rhizosphere. So for this, the next, I will answer. From the research, mortality is still low, not high you compare with Metarism or Beveria bassiana. But we can use trichoderma not only control of insect directly, but also we can use indirectly. Before my research, trichoderma aspergillum applied for plant by using seed treatment and spraying of crop, and after seed treatment and spraying of crop can reduce of insect pest of Myzus persicae. So for suppressed population in the land, maybe we can sure the is only using trichoderma aspergillum. Maybe for this my student is degree doctor, we can use combination with Beveria, Metarism, and Trichoderma aspergillum can suppress of species of insect pests of corn, Spodoptera fugiperda, is highly when we use only one species of entomopathogen. So for this research, I think using Trichoderma aspergillum not effective higher compared with Beveria bassiana. Maybe using combined with another fungi. So for Paul B. Okay, Bu Ginting ya, saya baca di chat.
Yeah, okay, ma'am.
Okay, yeah, okay. I don't have the check of persistency of trichoderma, and maybe climate change also affect the trichoderma population in the land. But after that, after this research, we can use when we application of the crop in the land maybe affect the persistency of this fungi in soil or in the leaf of crop. Okay, thank you very much.
And the last question, ma'am.
Oh, masih ada ya?
From Ibu Mitfah from Brin. Based on the research finding, what is the potential or in percentage terms of the study fungus to suppress pest population in the field?
Okay, maybe from my research we can apply the trichoderma on the eggs by when we use trichoderma the periods of instar insect maybe onto the eggs, maybe using prospect for control of insect pests, because infection of the egg also on to infection of larva of insect by the eggs development of insect also decrease the population of insect pests in the field. When we can use for control of only the larva, maybe suppress of population in the hive, and maybe not yet. Yeah. Okay.
Okay, ma'am. Thank you for you answer the question. And still any question? Mr. Henry Bustama? Have question, mister? Not yet. Okay. All presenter in this room, still any question?
Recording
Okay.
stopped.
Because not have question more, so the session discussion we stop, and you can join to main room, and see you next moment.
Okay.
Thank you very much.
Thank you very much, Bu Sempurna.
Thank you, Ibu Ginting.
Ya, Bu, sama-sama.
See you next occasion ya.
Ya, ya, ma'am.
Bu Sempurnanya ke Padang lagi nih.
Ya, Bu.
Bu Sempurna ke Padang ya.
Ya, Bu. Baik, Bu. Kalau ada seminar, tolong
Iya.
diinfokan agar kami ke Padang ya, Bu.
Oke, Konda ketemu aja lah di Bogor.
Oh, iya, ya, Bu.
Bro.
Nggak lama lagi ada seminar di Bogor, ya, ya, Bu.
Oke, thank you. Terima kasih
Oke.
semuanya. Ayo.
Baik, Bu. Untuk best presenter nanti diumumkan di ruang utama ya, Bu. Terima kasih.
Terima kasih.
Saya akhiri Zoom-nya ya, Bu. Sampai ketemu semuanya sehat selalu.
Program Studi Proteksi Tanaman atau PSPT merupakan salah satu program studi strata 1 di Fakultas Pertanian Universitas Bengkulu. Visi program studi yaitu menyelenggarakan program studi bereputasi internasional pada tahun 2025 di bidang proteksi tanaman tropis dan ekosistem pesisir dengan fokus pada eksplorasi keanekaragaman hayati serta pengembangan teknologi pengendalian hama dan penyakit tanaman yang berbasis sumber daya alam. PSPT berperan penting dalam pengembangan ilmu perlindungan tanaman. Dalam 5 tahun terakhir, riset dosen PSPT berhasil menemukan 4 spesies baru kutu putih yang berasal dari Indonesia, serta 8 spesies lain tercatat sebagai temuan baru di Indonesia. Data spesies baru tersebut dapat diakses secara lengkap melalui laman indomelibak.com dan kini menjadi rujukan nasional maupun internasional. Temuan lain yang tak kalah penting adalah ditemukannya virus baru pada pepaya Kalina di Bengkulu. riset karakterisasi begomovirus ini dilanjutkan bekerjasama dengan National Chongqing University di Taiwan. Untuk mewujudkan visi bereputasi internasional, PSPT telah melaksanakan berbagai strategi penguatan tata pamong, tata kelola, dan jejaring kerjasama. Di antaranya, melalui program World Class Professor pada tahun 2022 dan 2023, penyelenggaraan seminar nasional dan internasional, kuliah umum bersama dosen tamu luar negeri, serta keterlibatan aktif dosen dan mahasiswa dalam forum akademik berskala nasional dan internasional. Pada tahun 2023, 6 dosen Universitas Bengkulu terlibat dalam program World Class Professor. Salah satunya adalah Profesor Agustin Zarkani dari PSPT yang menjalin kerjasama dengan Çukurova University di Turki, serta riset bersama University of Malaya di Malaysia. Dosen PSPT lainnya, Dr. Mimi Sutrawati, aktif menjalin penelitian dengan IPB University dan menjadi pembimbing pendamping bagi mahasiswa proteksi tanaman di IPB. PSPT juga mendorong dosennya untuk berkiprah di kancah internasional, seperti Dr. Henry Bustaman yang menjadi dosen tamu di University of Heng Samrin Teng Bong Mum, Kamboja. Sebagai pengakuan kualitas, PSPT telah terakreditasi International Akwin sejak 2022 hingga 2030. Saat ini, dosen tetap PSPT terdiri dari 14 orang, termasuk 3 profesor, 4 doktor, dan 2 dosen yang sedang menempuh studi doktoral di UGM dan IPB University. Kompetensi dosen didukung rekam jejak penelitian, pengabdian, dan publikasi ilmiah yang sangat baik. Dalam 3 tahun terakhir, dosen PSPT menerbitkan lebih dari 220 artikel ilmiah, hampir setengahnya di jurnal internasional bereputasi. Dukungan sumber daya manusia yang berkualitas juga tercermin pada rata-rata beban kerja pengajaran, penelitian, dan pengabdian kepada masyarakat yang melebihi standar nasional. Penelitian dan pengabdian dosen PSPT tidak hanya menghasilkan publikasi, tetapi juga luaran nyata seperti pendaftaran varietas tanaman baru dan buku ilmiah. Kemahasiswaan PSPT menunjukkan capaian yang membanggakan. Pada tahun 2023, terdapat 112 calon mahasiswa yang mendaftar dan 50 orang diterima setelah seleksi ketat. Hingga semester genap 2023 hingga 2024, jumlah mahasiswa aktif mencapai 194 orang. Masa studi rata-rata mahasiswa antara 4 hingga 4,88 tahun, dengan presentasi kelulusan tepat waktu yang melampaui rata-rata fakultas. Sejak berdiri hingga akhir 2023, PSPT telah meluluskan 89 orang lulusan dengan rata-rata IPK di atas 3,3. Sebagian besar lulusan bekerja pada instansi pemerintah, lembaga riset, perkebunan swasta, dan sektor perbankan. Bahkan, banyak yang menjadi wirausaha mandiri. Beberapa lulusan juga melanjutkan studi magister, baik di dalam negeri maupun di luar negeri, seperti National Chongqing University Taiwan. Dalam mendukung proses pembelajaran, PSPT memiliki fasilitas yang lengkap dan representatif, ruang kuliah modern, ruang kerja dosen, laboratorium riset dan praktikum, hingga Museum Serangga Kutub Putih yang menjadi satu-satunya di Asia Tenggara. Museum ini telah menjadi pusat rujukan peneliti dalam dan luar negeri untuk sabbatical leave, magang, dan analisis spesies. Laboratorium PSPT terdiri dari lab taksonomi serangga, lab nematologi, lab fitopatologi, lab pengendalian hayati, serta lab biologi molekuler yang mendukung riset hingga level genetik. PSPT juga aktif menjalankan program Merdeka Belajar Kampus Merdeka atau MBKM. Mahasiswa berkesempatan mengikuti pertukaran dengan 12 perguruan tinggi lain di Indonesia yang tergabung dalam APSITA, mengikuti kampus mengajar, dan berbagai kegiatan pengayaan pengalaman belajar. Penelitian PSPT diarahkan pada pemanfaatan keanekaragaman hayati sumber daya lokal dalam pengendalian hama dan penyakit tanaman terpadu. Selama 3 tahun terakhir telah dilaksanakan 74 penelitian dosen yang diseminasi dalam puluhan seminar nasional dan internasional. Sementara dalam bidang pengabdian kepada masyarakat, PSPT memiliki 2 desa binaan dan puluhan program yang terintegrasi dengan kurikulum pembelajaran. Capaian lulusan PSPT terus meningkat. Sebagian besar alumni berhasil mendapatkan pekerjaan kurang dari 6 bulan setelah lulus, dengan tingkat kesesuaian bidang kerja yang semakin tinggi setiap bulannya. Berbagai keunggulan PSPT antara lain kualifikasi akademik dosen, kurikulum berbasis KKNI yang relevan dengan MBKM, publikasi ilmiah yang produktif, hingga fasilitas laboratorium dan museum yang mendukung pengembangan kompetensi mahasiswa. Sebagai program studi yang terus berkembang, PSPT Fakultas Pertanian Universitas Bengkulu berkomitmen menjadi pusat pengembangan ilmu proteksi tanaman tropis yang bereputasi internasional, menghasilkan lulusan yang kapabel, adaptif, dan siap berkontribusi dalam pengelolaan pertanian berkelanjutan berbasis sumber daya hayati.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.
Ayo bergabung di Prodi Proteksi Tanaman Fakultas Pertanian Universitas Bengkulu.